BioHi-Fi, Professor Paul Neji-Keng, for giving us that insight so that we can all know where we are in Africa and where is AfricaBP going and how does it actually link to all other collaboration with the other institution. Let me now allow Dr. Isidore Hagai to come and give us the presentation on where we are also in terms of some of the genomics works that has been done across Africa with the collaboration internationally with the Roslin Institution.
Dear colleagues, good morning. I would like to thank UNISA and the AfricaBP leadership for giving me the opportunity to give this presentation. So I will first give a short background about diversity of African cattle, and then after that I will present key metric of the 5 cattle genome assembly. So in Africa, there is a lot of diversity of cat breeds in terms of phenotypic and genetics. And these have been doing, this is due to various events of introduction and migration. And we have about 150 breeds reported in Africa, and then representing up to 22% of the global cat population against 8% of European cat breeds. And these breeds are well adapted to various agroecological zones and resistant to disease. For example, you have some breeds adapted to the arid, semi-arid, and some adapted to tsetse belts, like Ndama, the West African Ndama trypanosauria breeds. However, despite the diversity of the African breeds, when it comes to reference genomes, they are very limited, especially for both indicas or zebu. And there is a recent study published by colleagues at the Roslin Institute where they built genome assembly for the Ndama and Ankole cat breeds and use that as a pan-genome graph, putting many reference genomes together, and they have noticed that it contains an extra 116 megabase, which is absent from the heifer reference genome by that time. And they have also shown that the graph genome increased read mapping and also reduce allelic biases and improve structural variant calling. So let me link genomics to bioeconomy, or how we can use genomics to improve productivity. Here I will give example of dairy cattle, for example, when we want to do genomic selection. So to do genomic selection, you need to genotype certain number of cows, for example, and also phenotype them. And now, after that, you link the genotype to the phenotypes and then develop prediction equation. Now, after having your equation, when you have the selection candidate, you just need to genotype them, and then you can estimate the genetic merits. And it has the advantage to select young animals, even from birth. If you genotype your animal, you can select them. And because of shortened generation interval, the genetic progress will be increased. And it is cost-effective compared to the progeny testing, where you have to measure performance of progeny before you select them. And with this genomic selection, we can improve productivities, increase incomes for farmers. And when the farmer is able to produce enough food, they can sell and even import, and that will contribute to the economic growth of the country. Genome assembly for 5 African cat breeds. So the first one is Bosuras Laguin, a triple breed from Benin Republic. And there is Gudali on the bottom left, also from Benin Republic, which was Indicas and known for its milk production. And in the middle, we have the Singida White from Tanzania, beef breed and draft animals. And we have the Mpapwa, which is also a breed developer in Tanzania. It is a crossbreed, dual purpose for milk production and beef. And we have also Ringared, beef animals, and also used in agriculture. So we sampled the animals and extracted the DNA and did a PacBio Hi-Fi sequencing on review platform at the University of Edinburgh. So from the raw reads, we got Hi-Fi yields ranging between 160 gigabase to 219 gigabase. And you can see that the coverage is very high. We had at least 50x. So after standard quality control, we assembled the genomes using Hi-Fi's. And we can see that for the 5 breeds, the genome sizes were higher than 3 gigabase. And we can see that they are highly contiguous. There was no gap, and with few contigs when compared to other reference genomes or assemblies. So when we talk about contigs, the higher the contig is, good it is. So the contig is like when you have your assembly, and then the assembly is made of different segments of DNA. When you order them from the longest to the shortest and start counting them, the length of the contigs at the 50% of that assembly, that is the contig length. And then when we are talking about L50, that is how many contigs or scaffold put together made 50% of the assembly. And after that, we try to see what are the fraction of the most recent reference genome or genome assembly. What is the fraction of that contained in our assemblies? And we can see that it ranges from 95.2% up to 96.4%, which is very important. So here I'm showing the mercury spectra. So that is used to assess the accuracy of our assembly. So for example, the gray area is the sequences shared between our raw reads and our assembly. And higher it is, higher the accuracy is. And you can see from the 5 breeds that the gray area is most important, with a small misness, like, so the green is the largest part, and that is the share between the sequence read and the assembly. And on the left, when you have the gray, that is reads only in the sequence, but not in our assembly. And the quality of our assemblies range between 67 to 71. When we want to talk in terms of percentage, that is higher than 99.9999%. So here we have the genome completeness of our assembly. And as you can see, the completeness is higher than 98%. And we have less than 1% missing genes or autologs. Now, when we want to compare to the two recent assemblies, the Ndama and Ankole, for example. So when we take the scaffold, the scaffold 50, we found that it ranged for Ndama and Ankole, it ranged between 84 megabase to 100 megabase, while in our assemblies, they were all higher than hundreds. So after that, we try also to see how our assemblies fit in the publicly available genome assemblies. So our new assemblies are in blue, and you can see that the breeds with Indicus blood, they cluster together. And our Bos-Thoras laguine cluster together with Ndama. So Ndama and laguine are two West African trypanotolerant breeds. So as conclusion remarks, there is a potential for future research to unlock more benefit from African cartogenomics, more genes to discover to improve productivity and climate resilience. And to achieve that, there is a need for continued investment and collaboration to realize these opportunities. And also there is a call to action for stakeholders to support genomic research for sustainable bioeconomy. So I can't finish the presentation without acknowledging the Royal Society that was under my Newton International Fellowship. And we sequenced the Laguine and Gudali breeds with that fund. And the Rosalind Foundation for two breeds from Tanzania, and GCRF grant we got together with Professor Apollinaire to sequence the Mpwapwa cat breed. Thank you very much.
Do we have any, just one or two questions for Isidore?
Yes, Isidore, those are very nice numbers for the genome assembly, so congratulations.
Thank you.
I was wondering if that was PacBio Hi-Fi only sequencing, or you actually had Hi-C in there as well to get the scaffolds. I'm wondering, how did you scaffold? Yeah.
Okay, it was only the Hi-Fi. So we did the reference scaffolding using the most recent HER4 assembly.
Okay.
And we use a RagTag for that.
Okay. Is it possible to make the Hi-C data on these same samples? If there's sample left over?
So we don't still have DNA for those animals, but some of them, for example, the Mpwapwa breeds, we still have the animals and we can go back and get sample, even the trypanotolerance. Only that there were no resources at that moment to do the Hi-C.
Okay, thank you.
Yeah.
Any question? No? Oh. If we don't have any question for Isidore, let's. I'm suggesting something here that we allow Professor Nicola Moda to actually give us the talk now before we break for tea. so that we can allow her also to go on another meeting that she would have this afternoon. So if that is okay with everyone, it should not take us long. So if we could just move into that talk and allow her to talk, and then after that, we're gonna take a tea break, and then we can then continue with all the talks as normal. So colleagues, let me welcome Thank you so much. Let me welcome Professor Nicola Moda. She's from H3 Africa, and they've been already doing a lot of work in terms of the human genome and strengthening the bioinformatics across Africa. And she will give us the overview from her talk on where we are. And let's give her a round of applause as she starts her presentation. Prof, I hope we are audible and you can hear us and you can share with us. There you go. If we can just give her the access so that she can unmute. Lucky, if you can unmute her. Are you able to hear us?
Okay, finally, I was able to unmute. I wasn't allowed to
now
unmute.
we can now hear you clearly. Thank you so
Thanks.
much, Prof.
Yeah, so sorry for the delay. Couldn't unmute, and sorry for jumping in before your tea break. But I have meetings from 11:30 till 6 p.m. and I was expecting to talk earlier. So I'm going to talk about the role of H3 bionet in capacitating bioeconomy, but actually it's more about, we haven't really got to the bioeconomy part, it's more about kind of the direct research side of things. So for a thriving bioeconomy, we need to have infrastructure for discovery and translation. And while people in the north might take that for granted, here we don't always have the infrastructure, the compute, the skills and everything to actually be able to do that, this discovery and then the translation. So we need those skills, we need industry skills, we need to have training in things that we don't normally have in academia, like how to think about entrepreneurship, innovation, and intellectual property. Then we need obviously the knowledge to be able to make novel discoveries and some way to have innovation and translation of that into some sort of bioeconomy. So if we look at the genomics example, I'm afraid the talk here is very much based on human genomics and health, because that's the area where we've been working in. But I think it'll come, I think there are a lot of parallels you'll find in biodiversity anyway. So we need kind of the capacity for generating genomics data, for handling samples, whether that be human or biodiversity samples, for managing the governance of those of the data and the samples. We need to have the tools for analysis and processing, so we can derive data knowledge from those. If we're going to translate that into something like medicines, we need to have additional information that's really relevant to our local context. And then we need to have kind of the infrastructure for managing the data at the end, in the end, and sharing and sort of allowing us to protect the IP. So the experiences that I'm going to talk about are through a few of the projects that I work on, so the H3Africa Consortium and obviously H3 BioNet, which is the big Pan-African network that we've been working on in H3 Africa. But I'm also now involved in the Harnessing Data Science for Health Innovation in Africa, DSI Africa Consortium, and I run the LYC Open Data Science platform for that. So I'll just bring in a few experiences from those. So H3 BioNet did a lot of work over the last 12 years in building the infrastructure for potential, bioeconomy, but really it's mostly for discovery, this side of things. So we built computing infrastructure, we built functional networks, we built some tools, we built training programs that allowed us to build capacity in all aspects of genomics. So from the sysadmins all the way to the endpoint of the people who are going to be analyzing and interpreting the data. And we got a lot of publications in terms of allowing for innovative research. Unfortunately, that ended in June 2024. So after 12 years, H3 BioNet is finished. I'll talk a bit later about where that's gone, but we do have a legacy. So we have a legacy of really increasing access to data capacity. So we built the genomics infrastructure because we knew this was challenging in most of the African countries. So this is infrastructure from data capture to data generation, moving and storing data, providing access to the data, curating it, QC, processing, analysis, interpretation, and then the downstream submission and sharing of that data. So we worked on tools for managing study metadata, clinical and demographic data, because we're working on human, the actual genomic data and other data. And of course, there was a very strong training component of this because we wanted to empower African scientists to analyze their own data and not have to move data off the continent. So one of the areas we looked at was sort of this managing of samples. People actually often forget how important it is to manage the collection of data around samples. And I'm sure in biodiversity, you understand how important it is to know, where was that sample collected from, on what date, what was the specimen, what were the conditions, etc. And so you have a whole lot of metadata around samples. And for human, when you're dealing with big cohorts, you need to think about collecting that in a harmonized, standardized way. So we actually developed sort of standard ways of collecting data. For humans, obviously, it was case report forms. And so we took all the case report forms from a whole lot of different experiments or different studies and tried to look at where these were collecting standard information and then develop standard case report forms so that any future studies would be collecting data about samples in a very standardized way. So in this case, again, it was clinical data. In your case, it'll be other data associated with your biodiversity samples. So we collected, we developed these standard case report forms, and these were for a whole lot of different diseases. And we put these into different formats. So we've got Excel spreadsheets for these, we've got REDCap templates for them, and we've got PDFs for them as well for these data collections. So we've got 11 data collection tools, 82 protocols. And many of these, although there were some from global cohort initiatives, we had to make some of the adaptations to make sure they were relevant to the African context. And so these are now all available on the GitHub page. Everything we do, we make open source, and these are downloadable. So this is something that I encourage you to do in the biodiversity space, is think about standard data collection so that I know you're working with different organisms, but that there's some commonality and standardization in this. And you can build tools using REDCap, for example. We also made sure we were working within the FAIR principles. So yes, the data, the samples, biodiversity samples, human samples, extremely valuable and precious. But that means that they're also very valuable to other people around the world who need to use them responsibly. So we needed to make sure that the data were findable, accessible, interoperable, and reusable. So we have a catalog where we have all the project data available, so you can, for searching, the metadata available, so you can search for the data sets you want and then actually request access through an access committee. All the data are the metadata we try to put into REDCap. We try to map these two ontologies so that they're interoperable. And then we provide information about what is the restriction on the samples. What can they be used for and what can't they be used for? And obviously for human, that's via the consent forms. For other studies, it would be via kind of the policy in terms of the sharing allowances for biodiversity samples. We also wanted to increase access by finding data sets that were already out there. So what African data, and in your case biodiversity samples, are out there that you can actually highlight and pull out of those databases. So they might be sitting in INSDC or some other databases, but you can make kind of some sort of, so what we did was actually make some sort of portal to highlight these samples so that if somebody's looking specifically at Africa, you could find them. So we have the African Microbiome Portal, which lists all the samples, all the studies that have been done on African microbiomes. And then we also have African Genomic Medicine Portal and an African Genome Variation Database, where we're pulling out information that exists in other databases, but making it more easily findable and kind of attributable to African scientists. So the Genomic Medicine Portal, for example, pulls out data from GKB, DisGeneNet, and other resources, because when the data is submitted to Africa to public resources, it's just submitted as African. Africa is, more than 50 different countries. You need to identify where was the sample collected, where was the study done. It matters whether it was done in North Africa, East Africa, Central Africa, or South Africa, because those populations are different. And so what we've done is curated these, so you can search now for different samples, and when you find the sample, it actually lists the country where the sample was, where the study was done, and then a link to the publication. So it's just highlighting the African data. And in this particular case, in terms of the bioeconomy, we were trying to make the data more usable in terms of potential use in genomic medicine. So how can you potentially use this information in the clinic? We also have the African Genome Variation Database, and this is just shown as an example of, yes, you can have African genomic data, but you also need to have African genomic data in a usable form. So just having CRAM, BAMs, VCFs, it's all very well, but how do you actually make this usable to the public? What does the public want out of that data? In the case of human, a lot of the disease researchers just want frequency data for variants. So in this, we actually provide frequency data that can be used for rare diseases. So for example, for a rare disease, you would do sequencing of a genome, and then you would filter your candidate variants by frequency information. And so we allow that to happen now at a granular level in African populations. So we don't give the data away, but we allow people to browse the data and search the data in a way that's useful to them for their clinical application. And this again leads to potential innovation and translation of the data. So the other way that we've really been kind of focusing on building capacity for bioeconomy and innovation and translation and knowledge is by building skills. And the genomics workforce, as I mentioned at the beginning, is very diverse. So we have our endpoint, who's a genomics researcher who takes the data, analyzes the data, and interprets it. But upstream of that, you have people who need to manage the workflows. You have people who need to manage the computational infrastructure on which the workflow runs. You have people who generate the data, who collect the samples, who collect the metadata, who clean the data. So we have an entire ecosystem of people that need to be trained in different areas. So we've tried to divide our audience into different groups. So the bioinformatics users, so those who are more the wet lab people and just need to dabble in bioinformatics. Then you have your bioinformatics scientists, who really are the core bioinformaticians who spend all day on the computer, and then the engineers who spend all day on the computer and are developing the algorithms or developing the workflows or setting up the computer systems for that bioinformatics user or the bioinformatics scientists. So it goes from sort of more technical side going up to the applied side. How would you use the data? And so we've run several different courses that tackle all of these different audiences. And we find that because a lot of the degree programs don't cover these fundamental skills, we do this through professional development. So either online courses or intense 1 week in-person courses. We're also thinking about the next generation of bioinformatics leaders. So it's all very well to train in skills applicable to a particular workflow, but how do we make sure that those scientists are going to become the future leaders? So we've actually managed to sort of through 12 years, we've seen some of our bioinformatics students go from PhD to postdoc to academic, all the way into independent academics. Some have got positions at many sort of prestigious institutions, both here and abroad. We don't like losing people abroad, but we do hope that they're going to come back. We also try to give people in our network leadership opportunities, so let them lead projects or co-lead projects and then become the leaders, have leadership positions in working groups, and then have a lot of networking opportunities. And through this, we've really managed to build a lot of new leaders, especially in our women. So a lot of the projects and kind of new initiatives are led by our women scientists. So the other way we try to increase capacity in a continent that is just so thirsty for knowledge and so thirsty for training, and so in dire need for training, particularly in bioinformatics, is we have this distributed classroom model where instead of training one group of 25 people for a week, we spread that out over sort of 2 to 3 months, and we train 1,000 people. So we have classrooms across the continent. We have people attending those classrooms in different countries, and then they're all linked to a single group where we have the lecturer, the training materials, and everything. And we've successfully run an introduction to bioinformatics course on this with more than 1,000 participants per year for about 7 years now. And we now run intermediate courses, so next generation sequence analysis courses, which are obviously more intense because now you have to run workflows. And so we create these virtual machines where you can actually have where you can have those workflows running locally. And the importance of that is when you go to a training workshop, yes, everything works beautifully because it's set up in that workshop. But what happens when you go home? So what we try to make sure is that those people who are on the course are using those facilities that they will use when they leave the course, when they finish the course. So they still have access to those workflows. And this is a way we're trying to sort of distribute the capacity to do next generation sequence analysis and other bioinformatics topics, of course. So our courses, I mean, they've just increased in capacity. We've reached people all over the continent. There's some gaps, obviously, where there are significant infrastructural challenges, but we have managed to reach a lot of people, and we've trained over 5,000 people with up to 50 different classrooms. So this has had a large impact of over 5,000 individuals, many of which have gone back to share their knowledge with other people in the institutions. Some have used that knowledge to complete their degrees or to get publications or to get collaborations. And one of the sort of small things we're doing here, kind of things that we can't quantify, but we find from anecdotes, is that it really makes a difference in people's lives because they started off with nothing. They started then doing their IBT course, managed to get some skills, and then went on to more advanced courses and are now actually not isolated people anymore. They feel like they belong to a community across Africa with long-term kind of support. So now that HRBAN is finished, we need to think about the next thing. So we're doing some training for the Africa CDC in the public health space. Africa Steering Committee wants to continue to still have some sort of activity. So we continue to support the catalog and the access committee. But we do a lot of our training now through the DSI consortium, so the Open Data Science platform, and a new grant called the African Genomics Data Hub, which I'll talk a little bit about. But these are ways that we're continuing to build capacity in slightly different ways. One is quite focused on human data. The DSI consortium is focused on data science techniques, which are basically an extension of bioinformatics. So the African Genomics Data Hub, we're building these web portals. We're building, trying to think about how we can have local copies of data, so local EGA, that's the European Genome Phenome Archive. We're even in talks with the INSDC, the sequence read archives, and the NCBI, the GenBanks and ENAs and that, about a potential African INSDC. We're also building other tools that work better for African genomes, and so sometimes the tools built in the global north are not necessarily applicable to our genomes. And so we're building more tools for that and continuing our training through this project. So these are just some of the projects. So we have tools and we have databases, and then we have a continuing help desk for anyone who has bioinformatics queries. How do we use this to build a bioeconomy? So I've talked more about the basic capacity for dealing with data and getting to a point of analyzing that data. But really what we want to do is think about how we translate that data. And that's something that's the gap that we haven't kind of got to yet. So one of the new initiatives we're looking at is building centers of excellence in genomics. And this is to respond to, our success during COVID in managing genomics data, but thinking about how we can get that to the next level. How do we take that data and get products? How do we take that data and get clinical implementations to really save patients' lives? And then we think the way to do that is to have a partnership between academia, industry, and public health, or some sort of government institution that is the outlet or the customer of the discoveries that you're making. So in the Genomic Centers of Excellence, we're requiring a partnership between an academic institute and a government led institute, in this case public health, and then connections to biorepositories, having support through a data core, but then direct connections to industry and to national government, because we want to sort of drive that innovation. And these centers of excellence, we work on 3 specific areas, including genomics workforce development. One of the projects that we're working on is actually a partnership with industry to sequence 25,000 African genomes, because this is something that industry can use to improve the medicines. And one of the things we insisted with this project is that those genomes all get sequenced on the continent. So the industry is basically come to the party and have agreed to put in some sequencing equipment. The first machines have gone into Nigeria, in fact, and to build a computing infrastructure so we can analyze this data on the continent, and includes a capacity development arm so that we can build capacity in the sample providers, so that the sample providers actually are empowered to analyze their own data. So we hope this will be a good pilot for working with industry on generating, analyzing, and sharing data. The ownership of this data stays with the African institutions. So still, even though, I've talked about all the work we've done over the last 12 years, we still have missing pieces. We haven't focused much on developing entrepreneurial skills. We do have sort of think tanks to try and develop kind of critical thinking in our trainees, but we haven't really done any training in intellectual property, in the real kind of shift towards building a bioeconomy. And that's where I think we really need to focus now. How do we move from discovery to innovation? And how do we build the capacity? So once we've got innovation, once we've got potential products, how do we ensure we have the capacity for manufacturing those products? So this is where I think we all need to focus now. So with that, I'd like to just thank everybody from the H3 Bionic Consortium and all the other consortia I work with, most of whom are funded by the NIH. And thank you for your attention. Sorry for keeping you from coffee.
Thank you so much, Prof. Nicola Muda, for the talk. And this actually gave us the opportunity now to see where we are in Africa in terms of all the projects that have been initiated and how far are we also in terms of the bioinformatics. I would just open for some questions from the floor and also online. If you can raise your hands, we'll be able to see you so that we can ask your questions. Please go ahead, Erich.
Yeah, so I wonder about how was ownership managed with funding coming from NIH, which is an American institution. How did they usually, if with NIH dollars, they say they with money being funded by people from taxes in the public, the genome sequences have to be available to the public. And that usually means the American public. So I'm just wondering, how do you manage that rules and regulation?
Yeah, so the data are available to anybody in the world, but they're available via an access, data and biospecimen access committee. So one of the requirements for the NIH and the Wellcome was that the samples and the data have to be made available to the global community. So, but the kind of concession they made was that there was a bigger timeline. So normally you would have to submit your data within a few months of generating it. They gave us 12 months, well, probably about 15 months to analyze our own data because we still had to build the capacity to analyze our data, and then a further 1-year publication embargo. So actually the data sets are all sitting in the European Genome Freedom Archive. They just, and then like all the other data sets in the dbGaP or EGA, they have to go via a data access committee. So in fact, they are actually available, but we had time, you know, a reasonable amount of time to use the data before they became public. For the PacBio project, that sequencing will all be done by industry. So we have ownership, but we will submit those to the public repositories as well once the African researchers have had a little bit more time to analyze the data. So yes, fully agree that these data sets are valuable and need to be global. And in fact, there are, as soon as the African scientists have had a little bit of time to use the data first.
Thanks.
So, Prof, you mentioned the issue of industry at least playing their role in terms of increasing the sequencing part within Africa, but I'm still seeing the lag from the government side. What are the things that you think we're still lacking, or as African scientists, we need to stretch to in terms of also bringing the government closer, be able to work with the government? What are the opportunities that you see us at least winning in terms of connecting with the government?
Yeah, that's a great question. That's a challenge we've had. You know, we've been trying for 12 years to get government on board to sustain these projects, and we have not managed. At every one of our consortium meetings, we have either, we've sometimes had the president of the country, we've had health ministers, we've had science ministers, and then they move on. So I think we need to get to the next level of government where it's not ministers that have a very fixed term and move on. We need to get to that level of government that, you know, have more stable positions. I think we can argue to government, you know, I think now's the time, given the close connection between the genomics academic groups and public health during the COVID response. I think government suddenly has a better trusting of scientists. Governments need to come in, I think, in terms of cutting the costs in import duties. So we find that, you know, we had to really, really convince industry quite hard to sequence here rather than overseas because it's cheaper overseas. And that's because of import duties on consumables and all the other challenges of consumables. So if governments can come to the party in terms of making it affordable to do genomics research on the continent, getting consumables in, supporting that. But they need to support the outputs. And we've, as I said, we've been trying for 12 years to do that, and we still haven't had success, but we will continue to try. And I know that AfricaBP has been really instrumental in trying to, you know, get the attention of governments. So we really just need to push this kind of trust they have in scientists now since the COVID response and say, look, it's not just COVID. We can use genomics for all other things that are beneficial to government. It's just having that audience. I think that's really challenging.
And thank you so much for that. The other thing that I see that we're lacking also is to keep our bioinformaticians around the continent. When I look at your graphs in terms of how many we're training, but if you look at now when one wants to recruit, you don't find them. Where are we going wrong? Or is it because we're not offering them good salaries to stay within the continent?
Yeah, I mean, it's really hard to find to fund positions for bioinformaticians and to fund them at a research, you know, reasonable level. So most grants will fund students or postdocs. And, you know, when we find that when bioinformaticians get into academic institutions in Africa, they end up having to teach, and then they just, you know, don't have much time for their bioinformatics. So we need to find sources of funding. We need to engage with the institutions to get them to recognize bioinformatics and bioinformaticians as core staff, so that we do have future permanent positions for bioinformaticians. And again, that's a big challenge. But also to have exciting projects. If as soon as we've got exciting projects and exciting data, that's how I keep my bioinformaticians. I can't pay them much at UCT compared to what they would earn in industry, but they're interested in the what matters in terms of the data, because it's interesting data for them.
Thank you so much. I don't see. Okay, there's one hand in the floor.
There is one question from the online chat. So this is from Tobias. Thank you, Prof, for the great presentation. I was part of the recipients of H3 BioNet on bioinformatics. How do I advance that to a degree? And
Thanks. I was just actually trying to reply to you. So the best way to do that is to think about what is the area that you want to go into. So people think I want a degree in bioinformatics, but bioinformatics is huge. It's structural biology all the way to proteomics, to genomics, transcriptomics. You know, it's a huge field. So think about where you want to be, what kind of area of specialization you want, and identify those courses. So we run NGS courses, we run microbiome courses or metagenomics courses. A lot of people around here run proteomics courses. So once you've identified the area that you want to go into, then focus on those skills. You can also sort of develop some more general data science skills, machine learning, statistics, biostatistics. Those are always good skills to have in programming. Basic programming, both basic Linux, and there are a lot of online courses for that. Keep an eye out on the H3 BioNet Twitter feed or the new projects, because we are always running courses. I think about the particular skill set that you want rather than just general bioinformatics and the underlying skills to get you there, like, you know, fundamental skills of a little bit of programming, a little bit of statistics, and then look for courses or identify PIs who are doing research in that field. Then maybe you can also learn quite a lot on the job. It's no need to necessarily go do another whole degree, because it's going to be quite a focused area. You can now do professional development training to get into that area that you want to be in.
Thank you so much. Okay. I would like to thank all the speaker. Prof, thank you so much for bringing us this talk and for engaging with us. I think there is a lot that as African scientists we still need to do. Maybe sit down and engage and see how best we can utilize the resources that we have and how best we can build the capacity. With all the audience, we do have other institutions that are joining us on Zoom, the University of Stellenbosch and also the University of Venda and also University of Northwest. So those are the universities that are streaming from their side. And then from here at ARC, we are streaming with the university around Gauteng. So we all welcome you. We did started later and we are behind in our program, but we're going to try to catch up with time so that we can finish the whole program. We are now going to break just for tea time at 10 minutes. I'm hoping that that is good enough for everyone. And then immediately after that 10 minutes, let's be seated and go on with the program of the day. And thank you so much, colleagues, for this morning.
could forgive you and see the children are starving and their houses were destroyed. Don't think they could forgive you. We were staying in Paris to get away from your parents, and I thought, wow, if I could take this in a shot right now, I don't think that we could work this out on the terrace. I don't know if it's fair, but I thought, how could I let you fall by yourself while I'm wasted with someone else? If we go down, then we go down together. They'll say you could do anything, they'll say that I was clever. If we go down, then we go down together. We'll get away with everything, let's show them we are better. Let's show them we are better. Let's show them we are better. We were staying in Paris to get away from your parents. You look so proud, standing there with a fountain and a cigarette, posting pictures of yourself on the internet, out on the terrace. We're breathing the air of this small town on our own cut class for the thrill of it, getting drunk on the past we were living in. If we go down, then we go down together. They'll say you could do anything, they'll say that I was clever. If we go down, then we go down together. We'll get away with everything, let's show them we are better. Let's show them we are, show them we are, show them we are, show them we are. Let's show them we are better. Let's show them we are better. Let's show them we are, show them we are, show them we are, show them we are. If we go down, then we go down together. They'll say you could do anything, they'll say that I was clever. If we go down, then we go down together. We'll get away with everything, let's show them we are better.
It's the most beautiful time of the year. Lights in the streets spreading so much cheer. I should be playing in the winter snow, but I'ma be under the mistletoe. I don't wanna miss out on the holiday, but I can't stop staring at your face. I should be playing in the winter snow, but I'ma be under the mistletoe. With you, shawty with you, with you, shawty with you, with you, under the mistletoe. With you, shawty with you, with you, shawty with you, with you, under the mistletoe. Everyone's gathering around the fire, chestnuts roasting like a hot July. I should be chilling with my folks, I know, but I'ma be under the mistletoe. Word on the streets and it's coming at night. Reindeers flying through the sky so high. I should be making a list, I know, but I'ma be under the mistletoe. With you, shawty with you, with you, shawty with you, with you, under the mistletoe. With you, shawty with you, with you, shawty with you, with you, under the mistletoe. Hey love, the wise men follow their star, the way I follow my heart, and it led me to a miracle. Hey love, don't you buy me nothing, cause I am feeling one thing. Your lips on my lips, that's a merry, merry Christmas. It's the most beautiful time of the year. Lights in the streets spreading so much cheer. I should be playing in the winter snow, but I'ma be under the mistletoe. I don't wanna miss out on the holiday, but I can't stop staring at your face. I should be playing in the winter snow, but I'm I'ma be under the mistletoe with you. Shawty with you, shawty with you, under the mistletoe with you. Shawty with you, shawty with you, under the mistletoe. Kiss me underneath the mistletoe. Show me, baby, that you love me so. Kiss me underneath the mistletoe. Show me, baby, that you love me so.
I should have stayed with you last night instead of going out to find trouble. That's just trouble. I think I run away sometimes whenever I get too vulnerable. That's not your fault. I wanna stay the whole night. I wanna lay with you till the sun's up. I wanna let you inside. Oh, heaven knows I tried. I wish that I could let you love. Wish that I could let you love me. Wish that I could let you love. Wish that I could let you love me. So what's the matter? What's the matter with me? What's the matter with me? I wish that I could let you love. Wish that I could let you love me. I wish I. And every time it gets too real, and every time I feel like sabotaging, I start running. And every time I push away, I really want to say that I'm sorry, but I say nothing. I want to stay the whole night. I want to lay with you till the sun's up. I want to let you inside. Oh, heaven knows I tried. I wish that I could let you love. Wish that I could let you love me. Wish that I could let you love. Wish that I could let you love me. So what's the matter? What's the matter with me? What's the matter with me? I wish that I could let you love. Wish that I could let you love me. I wish I wanna stay with you till the morning. I wanna lay with you through the sunrise. I wanna show you that you're my only. I wanna lay with you till the sun's out. I wanna stay with you till the morning. I wanna lay with you through the sunrise, through the sunrise. So heaven knows I tried. Wish that I could let you love me. Wish that I could let you love. Wish that I could let you love me. So what's the matter? What's the matter with me? What's the matter with me? I wish that I could let you love. Wish that I could let you love me. I wish I. I'm a big, big girl in a big, big world. It's not a big, big thing if you leave me, but I do feel that I will miss you much, miss you much. I can see the first leaf falling. It's so yellow and nice. It's so very cold aside, like the way I'm feeling inside. I'm a big, big girl in a big, big world. It's not a big, big thing if you leave me, but I do feel that I will miss you much, miss you much. Outside, it's now raining, and tears are falling from my eyes. Why did it have to happen? Why did it all have to when I open my eyes, you're gone. I'm a big, big girl, big, big girl. It's not a big, big if you leave me. But I do feel that I do. Is you mine? Is you mine? I'm good, yeah, I'm feeling alright. Baby, I'ma have the best freaking night of my life. And wherever it takes me, I'm down for the ride. Baby, don't you know I'm good, yeah, I'm feeling alright. Cause I'm good, yeah, I'm feeling alright. Baby, I'ma have the best freaking night of my life. And wherever it takes me, I'm down for the ride. Baby, don't you know I'm good, yeah, I'm feeling alright. You know I'm down for whatever tonight. I'll be the finer things in life. No matter where I go, it's a good time. Yeah. And I don't need to sit in VIP. Middle of the floor, that's where I'll be. Don't got a lot, but that's enough for me. Yeah. Cause I'm good, yeah, I'm feeling alright.
Taste 1, 2.
I'ma have the best freaking night of my life. And wherever it takes me, I'm down for the ride. Baby, don't you know I'm good, yeah, I'm feeling alright. So I just let it go, let it go. Oh na 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 na, no I don't care no more, care no more. Oh na 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 na, so come on, let me know, let me know. Put your hands up, na na na, no baby, nothing's gonna stop us tonight. Cause I'm good, yeah, I'm feeling alright. Baby, I'ma have the best freaking night of my life. And wherever it takes me, I'm down for the ride. Baby, don't you know I'm good, yeah, I'm feeling alright. Cause I'm good. Kiss me up before you go. Summertime sadness. I just wanted you to know that baby, you the best. I got my red dress on tonight, dancing in the dark in the pale moonlight. Done my hair up real big, beauty queen style, high heels off. I'm feeling alive. Oh my God, I feel it in the air to lift my eyes above. love. I sizzling like a snare, honey, I'm on fire. I feel it everywhere, nothing scares me anymore. 3, 4, kiss me up before you go. Summertime sadness. I just wanted you to know, baby, you're the best. I got the summer.
Good morning. Thank you. The music stopped, so I guess that is our cue for the second session of this morning's program. We have 3 keynote speakers and then 2 young scientist speakers in this session. Our first speaker is Professor Gill. Professor Gill is a professor in genomics at Texas A&M, and she's also a member of various interdisciplinary programs in genetics and biotechnology. Apart from that, she's also the principal investigator of the McGregor Genomics Project, which maps genes for production efficiency in cattle. So that's just a little snapshot from her CV. Yes, everything keeps on coming back here.
Yeah, I think the speakers on the stage, the volume needs to turn down, and that's what's giving the
Like
feedback.
so. I don't know. Is that better? Oh, those ones. Well, at least that one hasn't given any problems. So I'm sure that will be fine. And Professor Gill, with that, I give over to you. Good luck.
Hopefully this one's fine. Is that good? All right. Well, first of all, I'd like to thank Prof. Marpoli for inviting me. Still feedback? Still feedback. Now it should be good. Better? Okay. So thank you, Prof. Marpoli, for inviting me, and thanks to Lucky and Sinabongo for their arrangements that getting me here safely. And I'm getting a lot of feedback as well. Okay. Yeah, it's those ones. How's that? Still the same? Okay, I'm going to go. Just bear with me because I can hear my thoughts coming twice. So I was asked to talk about animal breeding and genetics for the bioeconomy, and you're going to hear a lot about genomics, and you already have. So I wanted to step back and share with you the history of cattle breeding in Texas. And before we get there, in Texas, we're entering the fall. And the fall is a massive season for a different kind of economy, and that is college sports. College sports drives much of the economy of our institution at Texas A&M. You want me to keep going? How's that? Use this speaker. Okay. Hopefully this is better. I can hear myself. All right. So in the fall, we begin college football. And for 12 years, we haven't had in Texas the biggest Texas rivalry, which is between Texas A&M and the University of Texas. And so this Thanksgiving, they'll finally come back and play. Now, our mascot is Reveille, the dog. And the University of Texas's mascot is Bevo, the longhorn. Now, the apocryphal story of how Bevo got his name is that some Texas A&M students in 1915 branded him with the score of 13-0 that Texas A&M had won the year before. And so they converted that 13 into a B, the dash into an E, inserted a V, and then the 0 became the O of Bevo. But that's the story in Aggie land. That's certainly not the story that University of Texas uphold. But that leads me into the story of longhorns in Texas and why they are so important to think about when we think about the bioeconomy of Texas as a whole. And there weren't always cattle in the Americas. Cattle were introduced into the Americas with Columbus's second voyage in 1493. So he provisioned his ships. There were 17 ships and 1,200 men on the second voyage. They were provisioned in Spain, and then they stopped again in the Canary Islands before finally landing on the island of Hispaniola, which is marked here. The capital is Santo Domingo. The island was also known as Santo Domingo at that time. So this is where Haiti and the Dominican Republic are now. So either in Spain or on the Canary Islands, he picked up cattle, sheep, and horses and brought them to the Americas. We now know from looking at some haplotype analyses of longhorns that it is unlikely that any African Taurus breeds contributed from the Canary Islands, and it was the Andalusian breeds from the Iberian Peninsula that formed the foundation for the longhorns. So between 1493 and 1512, there were more and more shipments but the population of cattle exploded on Hispaniola. And by the 1520s, they didn't need to import any more cattle. And indeed, they were beginning to export them from the Caribbean islands to Mexico. In Mexico, there weren't any fences, and some cattle got away. And so they spent about 80 to 200 generations semi-feral, adapting to the climate of Mexico and adapting to the pests of Mexico. By the early 1800s, there was the need for cattle, and so early settlers began rounding those up, and they had massive cattle drives. So here's a depiction of one of those. These were hundreds of miles long. They were driving them north from what is now Texas up through the United States. So the longhorns, you can see, they weren't particularly fleshy, but they did survive these sparse areas, so sparse forage, high pest load, and they were very well adapted to drought. The challenge for these early cattle producers were that these cattle had become so well adapted that when they interacted with breeds that were being introduced into the northern United States that hadn't been exposed to some of the pests that these cattle were actually carrying with them, there were massive mortalities. So the longhorn cattle had become immune to the tick, the Texas fever tick that carries Babesia. And if cattle have not been exposed to Babesia, then they will, now it's perfect, right? Then they will, then they will get these massive mortalities. So in terms of the bioeconomy, the cattle were moved up all the way through Texas into Oklahoma, up into Nebraska and Kansas. Ooh. And as the mortalities began, fences began going up. Northern producers began fencing off massive areas of land to stop these cattle drives going forwards. The railroads actually built trunk lines to come to meet the cattle. So we again had new parts of the economy forming. But eventually, there was a recognition that as railroads come along and can move cattle, longhorns really aren't that amenable to being loaded onto railway carriages, right? And so there was, instead of breeding and genetics to mitigate that issue for the longhorn breed, there was a wholesale replacement of the type of cattle that were used. And within a period of 40 years, longhorns were actually a threatened breed. And now we had a large number of Herefords, known in Texas as Herefords, and other continental and European breeds were more popular, much shorter horns, or indeed even polled cattle. You can see there's a couple of polled animals, so hornless cattle, which are much more amenable to animal handling in an intense situation in enclosed spaces. And even though it was quite straightforward for breeders to select upon that polled phenotype because it's a dominant trait. So you only need one copy of the polled allele to have hornless cattle. It took over 100 years to actually identify the causative mutation for polledness, which is a structural, there are several structural variants near the centromere of chromosome 1. And because there's so little recombination in that region of the chromosome, once the mutation arises, and it is a complex structural rearrangement as well, so there can't be reversion, it's easy to get that mutation to spread throughout whichever breed. So amongst the Angus and some other British breeds, the Celtic polled mutation, which is a 1212 base pair, mutation is popular. And then amongst the dairy breeds, there's another structural variant known as the Frisian mutation. And in the yak and some Chinese breeds, there's yet another mutation, but all in this same particular region of chromosome 1. What's really fascinating is we still don't know what causes horns. That's just one trait, right? And when we think about cattle, what's important in extensive production systems like we have in the southern United States, like we have in Australia, and like you have here across Africa, the majority of cattle in the world are in the subtropical region. So over 70% of the world's cattle are raised in the subtropical regions. These regions, as Apollinaire shared earlier, experiencing much more climate issues in terms of extreme events. We have just come through a drought in Texas that reduced the cow-calf population by about 4%. And other countries are experiencing those issues as well. So as we think about what we need to be doing in terms of cattle breeding and genetics in the future, it's thinking about those traits that again allow that part of the agricultural sector to thrive. And in these regions of the world, so much of that has to do with being able to successfully breed in stressful environments. So in many places of the world, cattle producers are paid for the weight of their cattle. That's how we sell many of our cattle in the United States. That's a proxy for the quantity of meat that the animal will have, but consumers more and more, even in low to middle income countries, as salaries do increase and more can be spent on good quality protein sources, there is a desire for better quality meat as well. We've seen tremendous advances in feed efficiency in the last 20 years, somewhat driven by genomic selection, but not completely. And then the most important things for any beef cattle system is the ability of a cow to have a calf every year, because the most significant cost for producers is the replacement cost of the animal. So the age at puberty, it will control when she has her first calf, and then her ability to rebreed after that first season is also very important. Then added on top of that, ability to tolerate heat, those endo and ectoparasites like ticks. There are many endemic diseases. And then we're also seeing more and more emphasis from consumers on animal welfare traits, and polled would be counted as one of those, but also the temperament of animals for safe animal handling, and then more and more emphasis on mitigating methane production as well. So even in high income countries, these traits are difficult and expensive to measure for most producers. Most producers in Texas have fewer than 40 head of cattle. 93% of producers in Texas have less than 100 head of cattle. Okay, so even where we have 12 million cattle in Texas, beef cattle, most of the producers are small scale. So they don't have scales to weigh their animals. And that makes all of these traits more and more complicated to measure. And I make a point of this because in order to have genomic selection, you have to have reference populations of the traits that you want to select for. And it is difficult to do that even in the United States for many of these traits. So you have to have accurate record keeping for those populations where you can get data, and you have to be collaborative at scale. So we need to cooperate within country and across country. And Dr. Napoli, Professor Napoli was a part of a very nice study on tick tolerance with Apollinaire and collaborators in Australia, building a resource population to be able to look at tick resistance. And that can be done by many cooperating producers and also by collaborating universities as well. I think there's also an opportunity, and we were sitting talking about how excellent your cell phone technology here is, not necessarily the Wi-Fi technology, but how much can be done now using a cell phone. And even some of the newer phones have LiDAR technology, so you can do 3D imaging of animals just with your phone. Can we use that to start to develop algorithms as proxies for weight and other very interesting traits? I think you also need to be nimble. We can't go back to the days where we just wholesale replace a breed. There are so many excellent opportunities because of the biodiversity of breeds to capture that. And we need to be making sure that we retain those for the years to come. So as we know, it's genetic variation that shapes our phenotypes, and those traits are all an outcome of complex and interactive evolutionary processes. And as I shared with the example of the longhorns, organisms adapt to changes in their environment, and those changes are driven by mutation and natural selection. Many of those can be propagated by animal breeders via artificial selection, and now that artificial selection is relying on genomic selection, which is really, in the intensively managed breeds, tremendously accelerating the pace of genetic gain. So when you have a vertically integrated breeding system like there is in dairy and swine and poultry, they have seen huge gains. We're not yet seeing that in beef cattle or sheep, but I think that the opportunities are still there if we can develop substantial resource populations to be able to do the training for those selection algorithms. So just to share a little bit about what we've been doing for the last 20 years was to focus on cow productivity. We began this project the year before the bovine genome sequencing project started as part of a collaboration to make sure there were resource populations available. And we chose to use a divergent cross between Angus, a British beef breed, and the Nellore, which was originally from India, but is now a popular breed in Brazil. And these are divergent, so they're the two subspecies of cattle, and they're very divergent in many of the traits that we're interested in. And then also these crosses are known for massive hybrid vigor in the F1s. And we were interested in seeing how far that hybrid vigor progress through the generations. And so we've been crazy enough to actually make F6 cattle. And we've stopped because they are not very good. So after 6 generations, we've exhausted heterosis for sure. So we have a project now where we are looking actually at the mechanisms that are contributing to heterosis and characterizing across this population of several thousand animals now, the regulatory regions of the genome and especially enhancers. But we are able to track through these populations the breed of origin of the haplotypes. And we sequenced the founders of the 13 full sibling families that we made in the first generation, in the first cycle of this project. And one of the traits that we looked at was the cow stayability trait. So this is the ability of a cow to have 5 calves in 6 years. So basically she has not had any skips within 6 years. And in our study, which is a Bos indicus influenced population, we found a significant region on chromosome 5. And this region has come up over and over again in studies where there is Bos indicus influence in the population. So several groups have identified very similar regions, but only when it's across. So if you use a straight-bred Bos taurus population or a straight-bred indicus population, you do not see this region. And so there's some complementarity going on that we think is due to structural variation on chromosome 5 between the 2 subspecies. What we then went and showed, and this was work by Bailey Engel, she showed that actually what's driving this is the ability of that heifer to get pregnant and then to have a second calf. If she has that ability, she will reach that stayability goal. And so it's this same region of chromosome 5 that is driving probably age of puberty, but certainly that ability to get bred as a heifer. We were able to identify some haplotypes in this region that are consistent with the phenotype, but also on the left-hand side here, you can see that there's a very negligible linkage disequilibrium with these other haplotypes. And again, we think that this is because there is a structural variant right there in that region. So to finish off, historically, breed substitution drove changes in animal productivity in the United States. And I've seen that in other countries as well, and heard Dr. Huaga this morning talk about migration and introduction of breeds in African countries as well. And so we just got to be conscious as we move forwards in our efforts for there to be mitigated risk. We don't want to lose genetic variation for adaptive traits like heat tolerance and parasite resistance while we focus on other traits. It's going to take collaboration at scale to accurately record these traits, and especially in extensive production environments, in order to be able to implement genomic selection. And I think we also have to be cognizant that crossbreeding is here to stay because of the benefits of heterosis. So thank you all for that. This is Bailey who did most of the work that I shared today. And we've had funding from USDA and several of the genotyping companies as well. And I'd welcome any questions.
Let's see if this one also behaves. Yes. Thank you so much, Professor Gill, for a very insightful lecture. Any questions from the floor? There were so many things that we could focus on. Yes, please go ahead.
All right. Yeah, I guess initiating questions here. Can you hear me? Yeah, okay. So yeah, great work. I'm wondering, is there any kind of concern as there's more integration or inbreeding and so forth of these different cow breeds, and with the change in climate, that there will be a crash, let's say, in beef production, cow viability, and can you predict that from the genomic sequences where things are headed?
So right now, the effective population size of cattle is about 100, and yet there are still pockets where there's tremendous diversity. I think the biggest risk, and even today there is a real risk of pest. So I talked about the tick. There is still a quarantine zone across Texas and Mexico where you cannot move cattle to stop the spread of Texas fever tick. And we've seen in the last few years breakthroughs of that quarantine zone, and likely due to changing climate, right? And so there is a risk of population crash. People have not yet investigated where the immunity comes from and how we could perhaps create exposure to protect naive cattle. I don't know that anyone's looked at, can we predict a population crash though from the sequence? But it's an interesting thing to think about.
Olivia? Thank you so much for the talk. I just want to find out, looking at the PacGenome consortiums and looking at where we are now in Africa, trying to build the capacity and infrastructure for genomics. How do we, from the South African point of view, how this can benefit us with the international consortium on PacGenome, for us to be able to benefit from that? If you have seen what we have done so far, we haven't sequenced more number in one species. We actually just building the reference genome, but other countries has that. How do we leverage from that through those consortia?
Mm-hmm. So the PacGenome Consortium is open to everybody who's interested in contributing. And so I think that there's tremendous benefit of contributing genomes to it. So they're also focusing on capacity building, and making sure that we are able to produce the assemblies without necessarily relying on the previous references, right? And then I think you have the resources from the genomic standpoint to leverage into genomic selection. But you also have to collect the phenotypes. So you can't have one without the other if you want to drive that bioeconomy.
Can I just follow up? So I think for us, the challenge was the phenotype, the recording, like within the continent. We're still even struggling now with a bit of collecting the sample from one country to another. And like for the Africa BioGenome Project, it's getting samples all over Africa, maybe to other regions where they already have the instrument. So even the collection of the normal phenotype through our smallholder farmers, it's still a bit of a challenge. And I think that's where we have challenges of building proper reference population. And maybe going forward is that, what are the better ways? Although now there is a lot, especially ARC, it's going out there trying to get a lot of the smallholder farmers to record their data and become on the database. And some do, subscribe from the breeders societies, which then help that they can have a specific way of recording the data. But it's not so much, and that actually disadvantages in terms of building the reference populations also. And the other thing I think that makes it so difficult, and that's why now we're hoping with the sequence that we have been doing now, it's going to help us to be able to identify more variants from the SNP panels that we already have, the one that are already been out, like the 50K, the 150K, and the 800K for us to use. So I think as we go now, it would unfold, but we still have that challenge of putting together proper reference population and also getting the farmers themselves getting a proper records for us to be able to record that and do a proper genomic selection.
So that isn't just an issue for Africa. That's an issue for many places. The small producers in the United States are not recording data either. So I as you were speaking, I was thinking back to the quote that got shared earlier, that innovation is thinking about a challenge as an opportunity. And so I think that is an area that needs to be given focus to really drive in the agricultural space, in the animal agricultural space, what you're doing in the Africa BioGenome Project to really have that transformative impact. And yeah, it is going to be finding ways to collect those data accurately, make sure that people are collecting the same thing the same way, so have those protocols in place, and then be able to build up those reference populations.
I think there will be plenty to talk about over lunch as well. So thank you so much. I think in the interest of time, we will move on to the next speaker. Thank you, Pat. Our next keynote speaker is Professor Japie van der Westhuizen. This will be a virtual presentation. Dr. van der Westhuizen is the General Manager of SA Statboek and Animal Improvement Association. And in this capacity, as well as the previous decades that he has spent in the livestock industry in South Africa, he has made significant contributions to this industry. So I think we look forward to hearing his contribution on the application of animal breeding and genetics and genomics, specifically in the African bioeconomy. Thank you. Japie, just a moment. The audio on this side is not audible yet. Okay, it's a pre-recorded one, but let's just get the audio sorted out.
making things right between us. But now it's all good, babe. Roll that backward, babe, and play me close. Cause girls like you run around with guys like me till sundown. When I come through, I need a girl like you, yeah, yeah. Girls like you love fun and yeah, me do what I want. When I come through, I need a girl like you, yeah, yeah. Yeah, yeah, yeah. I need a girl like you, yeah, yeah. Yeah, yeah, yeah.
I think we will go on with the next presentation. If we can kill the music, please.
We spend the day trying to make things right between us. Now it's all good, babe. Roll that backward, babe, and play me close. Cause girls like you run around with guys like me till sundown. When I come through, I need a girl like you, yeah, yeah. Girls like you love fun and yeah, me do what I want when I.
Okay, so we are having a little bit of a problem getting the audio of the pre-recorded lecture talk through the system. So we are going to go on with the next keynote speaker, who I'm told is already online, and that is Dr. Fatima Parker. Dr. Parker is the Deputy Director of the Biodiversity Informatics Partnership and Cooperation section at SANBI. That's quite a mouthful. Part of her mandate is making biodiversity data available in support of science and policy and policy implementation, as well as growing the biodiversity informatics spectrum in South Africa. So I think with that kind of CV, she's ideally poised to give us a talk on how the Global Biodiversity Information Facility can serve the African bioeconomy. So let's see if we have her online, and then we will go on. It is another virtual contribution.
Good afternoon, Chair. I hope everyone can hear me. Good afternoon, colleagues. I'm Fatima Parker Ali, and I'll be speaking to you on the Global Biodiversity Information Facility. And thank you for the opportunity to be able to present to this forum. Can I check? Am I audible?
Yes.
Thanks so much. So in this presentation, I'll basically be doing an overview of this presentation. I'll touch on biodiversity broadly and the bioeconomy. I'll speak to the meeting about the Global Biodiversity Information Facility, the value proposition and economic value of GBIF, our role in the national landscape as SANBI GBIF, some strategic objectives, the data landscape, and the science policy drivers. So in terms of Africa's biodiversity, I'm just trying to adjust my screen. Yeah, so Africa is the second most megadiverse continent in the world, 3 megadiverse countries, including South Africa, the DRC, and Madagascar. So here, biodiversity plays a critical role in sustainable development, provides vital ecosystem services, and is one of our greatest regional assets. The benefits of biodiversity are crucial to key economic sectors. We've been speaking a lot about that this morning, to agriculture, forestry, health, and to providing solutions to sustainable development and poverty alleviation on the continent. The management of our natural assets and information is therefore really critically important. In terms of our natural capital, basically Africa's natural capital and ecological infrastructure is very important. In South Africa, we speak about ecological infrastructure, which is nature's equivalent of built infrastructure. So this includes our mountain catchments, rivers, wetlands, coastal dunes, and is increasingly being recognized as important for service delivery. A significant proportion of South Africa's wealth is based on natural capital, and this includes both abiotic and biotic resources such as minerals, water, timber, fish, and our species. Our natural assets are also important to move towards a green and a blue economy, resulting in improved human well-being, social equity, while also significantly reducing environmental risks and ecological scarcity. South Africa therefore aims to transition to an inclusive bioeconomy, combining economic development, social progress, and environmental preservation, and have set ambitious targets to reduce inequality and greenhouse gases in support of our National Development Plan. But central to realizing the blue and the green economy are the conservation and sustainable use of our biodiversity and ecosystem services. So here producing reliable, relevant, targeted biodiversity data will support better and targeted decisions by policymakers and leading to smarter economies. So the collection, collation, digitization, preservation, and dissemination, as well as the use of biodiversity data, are therefore critically important. So while it's very positive that economies have grown in Africa, and with South Africa's transition to a green economy, we do need to ensure that the data that supports our analysis are being mobilized and the priority data sets are available to support analysis for, amongst other things, natural capital accounting, our different sectors such as energy, climate, water, food, as well as trade and investment. So research infrastructure such as GBIF then enables this. GBIF was basically established in 2001 as a mega science initiative and is an intergovernmental organization composed of many national nodes. South Africa is one of those nodes, as well as international organizations. Collectively, and you can see on the image below, collectively, these there's over 100 country nodes as well as international organizations that are members of GBIF, as well as many international organizations that also publish data to GBIF. So the GBIF research infrastructure has become an enabler for the data science policy value chain and supports effective decision-making. The aim here really is to mobilize and to put to use vast amounts of biodiversity information, thereby advancing scientific research in areas like agriculture, health, conservation, and to serve the economic and quality of life interests of society. So in the current context of South Africa then also, in the context of the Science, Technology and Innovation White Paper, South Africa has become a member of GBIF to strengthen its efforts in open access philosophies and to drive towards a knowledge economy. So here, data forms the basis for innovation. Economic growth is driven by innovation, and the key driver for innovation is high-end human capital. So the development of capacity, therefore, we've heard earlier through other talks as well about the critical importance of capacity development. So once again, the development of capacity in biodiversity informatics, supporting open science is one of our key focus areas as well. Currently, GBIF has mobilized approximately 3 billion occurrence records. And this we do through its many country nodes, through more than 2,000 publishers, which includes international organizations as well as country nodes, more than 100,000 data sets itself. And a lot of this information is being downloaded and is also being used for publications. The as I've mentioned, the data is being used, and in this slide, you'll see that it's increasingly being used over time. And currently, more than almost 2,000 peer-reviewed articles have been compiled. And an interesting publication then is the GBIF Science Review, which is basically a summary of different kinds of research outputs using GBIF-mediated data. But on the bottom diagram, you'll also see that Africa also needs to increase its outputs in terms of research using this GBIF-mediated data. So if we move on and if we think a little about the origins of GBIF, GBIF's origins is through the OECD. And initially, it's the first phases focus very much on mobilizing low-hanging fruit, looking at our natural history collections, mobilizing plant specimen records, as well as insect biodiversity records through our museums. But through a process over the last 20 years, let's say, it's moved from simply just mobilizing low-hanging fruits, lots of bird biodiversity data records, towards also a more mature approach where it's now also able to look at impact and really tailor its approach also to ensuring that not only are we mobilizing data, but ensuring that this data is relevant. A lot of, as I've mentioned, there's a lot of uses of the data, the science review being a key output where the scientific community can also engage with that to see what type of research is happening. But some of the key outputs also is that some of the data has found its way also into the IPCC report. And GBIF also, in terms of its value, it's also directly contributing to the global biodiversity framework outputs and to indicators within that. And those occurrence records then contribute to many of these indicators, such as species conservation, invasive species occurrences, and so forth. So there's a clear policy outcome and clear policy importance in terms of the data. In terms of the economic value of GBIF as well, there's a very interesting publication that has been produced, the Economic Valuation Report, which basically indicates that for every euro invested in GBIF, there's $3 of benefit to the user, up to 12 euros to society. And basically here we're talking about the value in terms of, so 15 million in terms of the investment, financial value of GBIF is how it's been identified. And the access then, increasing then to the user in terms of the access and time saved. So the user not needing to go out and collect information, but the information being relevant and available on your desktop. And then approximately up to 185 million value in terms of research impact. So this is a clear value proposition then also of GBIF. At the national level, we have a number of key strategic areas that we look at supporting, which is to grow human capital in biodiversity informatics. And here we try to engage with academia, as well as we offer training and learning approaches as well. In biodiversity informatics areas, we also look at growing the community through partnerships around data mobilization, engaging in strategic national and international regional initiatives, and then also to promote the mobilization, use, and awareness raising of GBIF. So in terms of capacity development, we aim to have a holistic approach while engaging academia, also providing training activities. And then we've also been, as I've mentioned, engaging with academia towards a conceptual approach to developing biodiversity informatics nationally as well. And there we've basically looked at working with some of the universities, and we've produced a publication around that. We've also engaged in terms of developing curricula in terms of growing the field of biodiversity informatics. We've worked with some of our GBIF partners through the National Nodes network, and we've looked at a curriculum for biodiversity informatics, which moves across the value chain of data generation to data management, biodiversity data management, to its use and application. And we've then also developed a portal to showcase the data that we do have. We've mobilized more than 37 million biodiversity records. And when I say that we've done that, it's our partners across the country, our network of partners that has mobilized this information. So it's not just one organization, but rather the network of universities, research organizations, museums, and so forth. So as I've mentioned, we've grown to mobilizing more than 37 million currently. A lot of this being observation-based records and a lot of this being bird data, but also a substantial amount of our plant information as well, our specimen records that's available on GBIF. We enable this data through what's called the Integrated Publishing Toolkit. So the data sets are mobilized through our partner organizations. We use the IPT, what's called the IPT, to enable the dissemination of our national level data into GBIF. And we are currently also, we've recently initiated a conveyor belt system for mobilizing data at the industrial scale. And this is specimens from the Pretoria Herbarium that's shown here that we've imaged. So currently the setup is in Pretoria and will be moved, but this system really does allow us to accelerate the process of imaging natural history collections by orders of magnitude. So really scaling up our digitization processes or our imaging processes for natural history collections. Further to this, in terms of our training as well, we support the improvement. We try to ensure the improvement of data quality and to enhance use and uptake of data. And so we've therefore have training programs that we do make available or training workshops. And we've had a number of them over time. We try to have a few of them annually. And this information is available on our website. And it's accessible through a login. Our partnerships, we try to also engage with partners both locally as well as internationally, and we've been engaged on a number of projects. Recently with our Norwegian colleagues, the GBIF node in Norway, with regards to accelerating biodiversity data, also training workshops and ensuring and also enabling some studentships along the way. In terms of the data that we are mobilizing, what we are really looking at is when we're talking about our occurrence records, specimen records, literature, animal tracking, observations such as the iNaturalist type of information, or sequence information. So really what we are looking at here is what type of species, where is it found, when was it found, what's the evidence, and as we've mentioned, specimens or sampling event data or observation records. So in terms of richness, and I'm sure our communities are familiar with this, we at the most basic level, we're talking about metadata, the type of data that's supported by GBIF, occurrence species checklists, which is species in countries or particular areas, species with dates and coordinates, and then at the most rich level, we're talking about sampling event data, which includes species with dates, coordinates, methods, abundances, etc. And more recently also, GBIF has looked at mobilizing barcoding data, eDNA records, and so forth. And this really is to also support or correct the taxonomic bias, which most of us are familiar with. And there are a number of resources also that the community can look at to enable DNA level data to be mobilized. There's a guidance document, and this is very recent work that GBIF has been looking at to enable some DNA data also being mobilized. There are other initiatives as well, where GBIF are partnering with a number of those international initiatives, but also they are looking at mobilizing some data. And there's also a tool that's been made available, although this is still in its very early stages. Yeah, but there's more information available in that guidance document. So in terms of the policy landscape, there's a number of policy drivers which support biodiversity informatics broadly in the country. There is the Southern African Research Infrastructure Roadmap, the National Big Data Strategy. Core for the node as well is the White Paper on Science, Technology and Innovation. And then also for SANBI, we are also the key organization that's one of the organizations also in producing the National Biodiversity Assessment. So that's also quite key for our outputs in terms of the CBD. In terms of the programs supporting biodiversity informatics, we have the Foundational Biodiversity Information Program, which is a long-term program to generate, manage, and disseminate foundational biodiversity knowledge to improve decision-making, service delivery, and creating new economic opportunities. So this program has a clear policy linkage, so very clearly important for our bioeconomy and for growing our bioeconomy. Several large projects have been funded through this initiative. And an interesting one is the SeaKeys initiative. And I mentioned this one because it had a very, very clear, interesting outcome in terms of supporting the policy linkage. And here we're looking at the data that was mobilized through this network, which included specimens, it included barcodes, it included molecular information, species checklists, and so on. But basically what they were able to enable was an increase in the marine protected areas and an increase of 0.5% to 5%. So that is quite noteworthy. And then maybe perhaps to mention also that through the GBIF network, we've also been engaging in the Research and Innovation Working Group of the G20, where Brazil has basically been brought GBIF into also support aspects around species cataloging and expansion of open biodiversity databases. And so here we've also have a very clear linkage with innovation. We've in one of the priority topics, there's also the G20 support for open innovation and cooperation. And here we're also talking about unlocking the latent economic value of ideas and knowledge. So just a strong sort of push around innovation and the economy. So just to end off, in terms of biodiversity informatics and how this data supports our science and policy, at the base of it, we have different types of biodiversity data, such as images, citizen science records, specimen data, mobilized through different initiatives, such as the Foundational Biodiversity Information Program, iNaturalist, and these all contribute to global data sets that are then being used in scientific research for expert assessments and also into the science policy interface. This data it supports the science policy, but ultimately also this information is relevant for our national reporting and our processes, and in this way really does support the bioeconomy. So I think, yeah, so thanks so much to our organizers and for the invitation to present on GBIF. Thanks so much.
But we can now communicate to them.
Yes, yes, I had it on. Okay, we are back online. Olivia, you can go ahead with your question, please. I'm just going to allow this one question. Sorry.
Okay, so thank you so much for the talk. My question or my comment is on the samples that are collected that in future are used for molecular analysis or also for genomic analysis that we are accessing through SANBI. And I wonder how we can improve the quality of those samples, because we look at the samples that have been collected over the years that we try to test and see if they can be usable for sequencing, and we realize that they're not. So my question or my comment is, how do we improve our collection methodology and our storage methodology so that the samples that we are collecting now, especially because they're of importance, it's not easily to collect them or to find them. How do we also store them in a way that it's much safer and they will be usable for future use? Thank you.
Dr. Parker, are you still there? IT, is she still online? Okay, otherwise, Olivia, maybe if you can compile questions and email and so on. So we are now going back to Dr. van der Westhuizen's presentation. I have already introduced him, so I'm not going to do that again. Dr. van der Westhuizen is now joining us online for a live online presentation on the application of animal breeding and genetics in the African bioeconomy. Please go ahead, Dr. van der Westhuizen.
Good afternoon. I hope you can hear me, Chairperson. If you can just indicate to me whether you can hear me and see my screen.
We can hear you. Please go ahead.
And see my screen, hopefully.
Yes.
Okay, cool. Then I can go ahead. I've jumped through a few hoops to make the recording, but the recording is there afterwards, and I might have been better as an actor than a presenter. So hopefully you can follow that. It's really a privilege to be here today, and I would also like to thank my fellow my colleague Jason Redding that assisted me to a large extent. I'd like to kick off by if this thing can just move forward. There you are. Kick off by saying what are we trying to achieve in terms of quality animal agriculture? We'd like to unravel the phenotypic differences amongst animals, and I will concentrate on production animals or livestock. So we would like to know more about what's causing differences in response to production systems or environmental differences and so forth, and how can we assist in having the type of response that we really need in the livestock sector to be sustainable in our production. So we all know that basically we're talking about not just that variance, but also the variance or variation in genetic makeup of animals, and also their response on specific environmental differences or environmental restraints in terms of production. I'd also like to go back a little bit in time and say, what are we talking about? And if you look at these distinguished gentlemen over time, that had a marked impact on unraveling what's happening on the genome level, given the fact that they were in a time slot that less were possibly known about what we know today at the genome level. So Gregor Mendel, working on his the color variations in P's, basically said P resembles G. So what you basically observed in terms of color should be on the genetic level. Robert Bakewell, for those of you who know, is also known as the father of modern animal livestock breeding. He discovered the fact that genes ran in families, and he coined the phrase like begets like. So if you have two good parents and you mate them, you will probably end up with a good progeny as well. So that was Bakewell. And then one of my heroes, Charles Darwin, he basically observed that there's very small changes through selection over time, and that led to the fact that if you have due to these changes and the natural selection, you got to sort of adapted animals and even species over time that's well adapted to specifically environmental constraints, or where the E changed, only those that were suited best for the environment were the phenotypes that ended up. In the more modern era, if we look at Fisher and Haldane and Wright, I think they assist us in understanding mutations better and how to use statistical means to describe that, and also to work towards probabilities that our decisions in terms of selection will end up with what we like it to be. J. Lush, where Bakewell said like begets like, J. Lush said it's not always the case. So although you have an expectancy in terms of family selection, there's also within families there are differences, and those differences need to be exploited in selection programs. Charles Henderson, and this is my phrase, not his, would probably said something like begets like to some degree. And that's why we need to include the relationship matrix in our prediction of genetic merit, and also those differences that we do record within contemporary groups. Not all of that is being transferred to the next generation. And then that's why we also have to include the proportional additive variance, or the additive genes, how much of those variation is caused by additive gene action, and meaning that the phenotypes that we observe will not be transferred 100% to the next generation. Also that we have to measure within so-called contemporary groups, or giving all animals an equal chance to perform in a specific contemporary group, and also the solving of the equations, the so-called BLUE, or the best linear unbiased estimates of the environmental differences that these animals had to perform in. Okay, so, and I'd also like to take a step back to say what's still happening, and how important these type of assessments are. If you talk to farmers, they still like to look at their animals. They still want to visually assess the animals. And so visual appraisal or assessment is still playing a large role in animal breeding and genetics. So it also sometimes acts as a so-called first line, or a gatekeeper, in determining whether you have the right animals for the right environment. And it also assists in some more refined genetic assessment But there's always, as I think as an animal scientist or animal geneticist, the challenge of telling them not always to link these visual assessments to objective recordings, rather take the next step of objective recordings. And then in many cases, it still also is being used as an assessment for the identification of unwanted genotypes that you don't want to farm with. Then pedigree information, and I think this has been the cornerstone in the seed stock industry for many years. If you can go back decades. If you take examples of horse breeds and so forth, especially the British were known for recording of pedigrees, and maybe that stemmed from Bakewell's comments in terms of family selection, and it also, there's a very neat structured systems applied, even naming animals in some cases. And it is interesting when the recording of performance really got the grip in the industry. Some people said, why still record pedigree information? And luckily we did not stop doing that, because we really need that in modern day genetic merit predictions. So, and we've also found that pedigree information still serves many scientific purposes. And like I said, we do incorporate them in genetic merit predictions. And it also remains one of the cornerstones of blood breeding value predictions, since we cannot genotype all animals in populations. There are few exceptions, like the Irish, the ICBF, genotyping over 1 million animals a year in a population of only 3 million cattle. So they basically take the bold step of just genotyping everything that's born. And that makes a huge difference in your accuracy of prediction. But pedigree information also assists us in having breeding strategies to curb excessive increases in inbreeding and loss of genetic diversity. And there are very smart mating programs out there that assist breeders to just that. And we also pick up that through that the frequency of single gene markers. By having the full pedigree, you can at least predict whether a new generation of animals might be carriers or not. And then also other important patterns such as retention and culling rates and so forth, we can use with the pedigree information. Then productive objective production recording. I said maybe since the 1970s it really took hold, but I think maybe a bit earlier. If you look at milk recording systems earlier on in the last in the early 90s, and you can see that people started using production recording. Obviously, the whole thing is that these recordings need to be within contemporary group, because otherwise you get a skewed picture of the real, if you use mixed model equations for the prediction, you might get a skewed picture if that's not done properly. Okay, I have mentioned the start of when Charles Henderson started the BLUP equations, and which then meant a very successful combination of pedigree and performance, the heritability or the additive variance and the covariance amongst traits to predict the genetic merit. And that was the first successful combination of like begets like and like doesn't always begets like in terms of the prediction of the genetic merit. The problem now, okay, this is just, sorry, a depiction of what goes into the BLUP equations and what's needed from a farmer's perspective, pedigree information and measurements. And then as a scientist, we'll work out the population parameters for these. This is a simple example of a mixed model equation for best linear unbiased prediction of genetic merit. The problem, however, is that there are some especially very important cases where it's basically important or impossible to determine the Mendelian sampling when you want to predict, especially young animals. That's not yet measured. If you just think of a trait like longevity, or even the repeatable measurements of female fertility, for example, sex limitation, such as female fertility, that you can only measure on females, or milk production that you can only measure on females. Some traits might be expensive or difficult to record, and that means just a very small portion of the population is being recorded for that. And so in all the other cases, you would not know the Mendelian sampling. And then there are also traits that you can only measure on dead animals, such as carcass properties or meat quality. So something had to be done to assist farmers and breeders to make more accurate decisions in terms of genetic selection of their animals. So the nice thing is, in the meantime, after the Human Genome Project and the Bovine Genome Project and a lot of other genome projects, the development of the bee chip really changed the scene at this stage. And the uptake, I think one can say, for the last 20 years of getting a glimpse into the genome of farm animals assisted us in using these technologies together with the existing mixed model equations to predict where we couldn't predict in terms of the Mendelian sampling. So we basically used a single step today, so-called single step, after an attempt on so-called SNP-LAP, where the SNP effects of additive genes were included. The problem there is you needed very large reference populations, and I think there were also mistakes still being made in terms of the direct genomic values being applied without proper phenotypes being recorded as well. So basically, by using the single step where the A matrix or the relationship matrix are being replaced with the genomic relationships of the animals that are genotyped, and we basically moved from BLAP to GBLAP, or genomic BLAP, nowadays. So if we go back to this depiction that we had, you will see on the right-hand corner here that the adage of the genomic SNP information on the limited number of animals, where you then change your average relationship matrix with the real relationships of the animals, and then do a BLAP on that in one step and get the predictions from that. So if we go to the classical equation for genetic change over time or per year, where do we really get assistance by including genomic information in that? And basically, like I've mentioned, the pedigree information is updated, especially where you have half SIPs or lower. Through the randomness of additive genes being transferred to the next generation, you end up with a little bit of guesswork in the current relationship matrix. But if you add genomic information, you have the true genetic relationship of all animals that are genomically tested, and that enhances the accuracy of prediction tremendously, especially for young animals, where you recently worked on a parent average value. I think we also see more in terms of the variation in genetic merit. We do that better. The intensity of selection can increase because you can work with a younger group of animals, and obviously you're more accurate in your assessment of them. And then the real gain that we saw in breeds like milk records, dairy animals, is the tremendous lowering of the generation interval, because you don't have to wait for progeny tests in pools for sex-limited traits. And that made strides in the genetic gains for them. Could just why what are the current drivers? Why are farmers paying for genotyping their animals? And we found that to a large extent, the single gene markers are playing a huge role. They're confirming certain things. We pick up the gene frequencies in breeds or overbreeds. Also, the relation with other traits of economic importance. We found, for example, the DGAT genes relationship with maternal ability of dams. We start picking up these relationship with single gene traits with what we term to be additive genes in the past. Novel gene discovery, even with not whole genome sequencing, but where you use the bead chip arrays and you maybe not have a direct hit on a gene. When you look at your Manhattan plots, you can immediately see where these observations were made, where the regions are, and then pursuing it even further. Parentage, very important. Confirmation of parentage, and then the fact that we use so many more markers, we can also use the SNP arrays outcome on discovery of parents, which we couldn't really do on the microsat markers simply because it wasn't as discriminatory as the SNP markers in terms of discovery and then lineage. If you just look at the slide on the right-hand side and you see this 3-generation pedigree of this animal, you will see that the animal was genotyped, its sire and dam, or the sire was genotyped, the maternal granddam was genotyped, and the maternal grandsire was also genotyped. So when I'm a buyer of this bull, I know with that's the truth that I see in front of me in terms of the line that the animal was born in. Homozygosity, if we look at effective population size, if you look at the straight in the middle, you will see that the so-called breed relatedness, which is the next point. This animal, we've developed so-called breed keys, so that assist in upgrading systems, half crosses, 3/4 crosses, to really check what the relationship are, simply because of the randomness of passing genes from one generation to the next. You can check yourself on that. And then the level of homozygosity, this is fairly high, 30% in this case. But that by tracking that, one can also manage genetic variation in the future. And then GPLUP, I've mentioned that. Investment in genomic reference populations has become important, and then including these GPLUP in breeding objectives and mating plans. Just to give you a glimpse into the numbers of genotypes on Statboek's database currently. Since 2013, the Bonsmaras invested in a reference population, and we genotyped just under 1,000 animals. If you look at 2013 to 2015, then we were lucky enough to get assistance from TIA, the Technology Innovation Agency, for the first beef genomic program, and a lot of animals were genotyped then. That was followed by the DGP, the Dairy Genomics program, and then breeders started investing in genotyping their own animals. As the 1st of June this year, we had just had over 26,000 genotypes on our database, and we're looking forward now to the second round of the beef genomics program, where a multitude of animals will be genotyped, and also from the developing sector. And we're looking forward to see how much information we can really get out of those genotypes. These are some of the breeds already getting GPLUP. The red bars in this graph, you will see some have lower than 1,000 animals being genotyped, but we, through test, especially where you have a close-knit in terms of genetic relationships in a breed, it's possible to get away with single step and having less genotypes. The green ones are yet to have genomic selection or GPLUP being employed for them. Just a few interesting like breed analysis that comes out. This is the example, and it's been previously mentioned, the POLD mutations, Celtic and Frisian POLD variants, and it's possible if you look at the top is a Tuli example, basically a POLD breed, and you can see the homozygous POLD Celtic variant, and the carriers are making the bulk to 3/4 of the animals are carriers, either carriers or homozygous for that, versus the Boeran. Both of these are African breeds where it's mostly horned animals. So I think a lot of other information coming out of and this is on a commercial scale. So these guys pay for their own genotypes, and these are services that they render. On the meat tenderness side, if we look at the calpain and calpastatin frequencies, that's basically from muscle to meat, and this is an outcome of a project that was earlier held, and then the tenderness associated with this. And this is just the two, if you look at the round graphs at the bottom from level 1, which is less tender, to level 6, most tender, you will see a huge difference in gene frequencies between these two breeds, the Boeran and the Nanopore, in terms of the leaning towards more tender versus less tender meat in terms of the series of genes that was basically available on the SNP chip. I also think that genomic information in breed assessments frequencies, this is basically based on just over 9,000 genotypes, and immediately you can see how many parentage conflicts there are, how many the variants in terms of the Celtic variant, the Frisian variant, and then the unwanted double muscling genotypes and carriers of that. And things like I've mentioned, the DCAT and even the SLIC gene. In this case, it's a breed not known to carry the SLIC gene, and therefore none of these animals are really carriers of the SLIC gene. Also, I've mentioned homozygosity, the management of that, and outcrossing if need to be to recreate genetic variation over time, and also to manage genetic diversity in a breed. So one can at least, and these are birth years at the bottom of this graph, and the level of homozygosity on the y-axis. And then I have mentioned the unwanted or detrimental mutations, managing that and checking in what families it runs, and also managing that to get your breed basically free from unwanted mutations. I had to put this in, and I don't know how many of you have actually read this far in what Darwin has written. He was quoting Livingston, and I'm just going to read this. He said, "Interior African people cherish the good qualities of their domestic breeds, people that have not been associated with Europeans. Keepers of cattle show practices that support the theories that the breeding of domestic animals were carefully attended to in ancient times. It would indeed have been a strange fact had attention not been paid to breeding, for the inheritance of good and bad qualities is so obvious." So basically what Darwin has confirmed is that it's really ridiculous to believe that there were no selection amongst the indigenous Sangha breeds in Africa, and that they just came about merely as the result of randomized mating without the interference of people. So I think there's a good argument that the African herdsmen and the owners of cattle laid down so-called, in my words, breed standards by favoring some bulls as favorites to father the next generation of progeny. So I really think, and we can see this if we just look at the principal component analysis, and these are a few African breeds, and with two non-African breeds. On top is the Boeran, the African Indicus, And then the black cattle on the right is the Drakensberger. The Nguni is well known. The Senapol, which is a polled animal, and the Afrikaner on the left-hand side. The two European breeds or other breeds, European breed and another Indicus, the Brahman, the yellow dots there, and the Sussex, just as an example, to show you the distinct differences of the African breeds and the Sanger breeds with those of the continent, the UK, and also those of Indian or mixed origin in terms of Indicus. So I think there's so much scope for us in Africa to exploit this. Maybe exploit is not the right word, but to use this in our selection programs. Another very interesting example, and this is the merino sheep in South Africa. We also have a few thousand genotypes of sheep on our system. And this is different flocks within the merino. The green one on top is basically one of the foundation flocks with a closed flock approach. And you can see the variation with the other flocks. And this also assists us in to tie these flocks together with our genetic evaluations, where you basically need that as well. In closing, I think I just want to give the old perspective again. We're talking about genotypes and using genomic predictions and so forth. But you always have to have this perspective in breeding. The phenotype remains king, as Mike Coffey from Edinburgh University phrased so well many years ago. It needs to be well defined. You have to accurately measure them, and you have to have contemporary groups. And then you can build the next level, the so-called genetic merit predictions. I've spoken a lot about that. Then you have to have breeding objectives and use these predictions in well laid out breeding objectives that looks at the biological differences, the market and the resources available, and obviously the environment. And then you have to have optimal matings to achieve your goals and to retain the additive genetic variance in the breeds that you work with. That is my talk, Chair. I hope I haven't overstepped my time. My recording was spot on, but I tend to talk a lot, so hopefully you can live with my presentation. Thank you very much for the opportunity.
Thank you, Dr. van der Westhuizen. We'll have to trust you that your presentation, the pre-recorded one, was just on time, but you kept within your time limit. Thank you very much. Again, unfortunately, we are running a little bit over time. So one or two questions, please, and then we will move on to our young scientist talks. Any questions from the floor? Yes, please go ahead.
Yeah, same question I had earlier. I was wondering about in terms of using the genomics to predict future population bottlenecks or diseases and so forth. And I'm wondering, have you been thinking about that in your efforts as you pull all this data together?
Yes, as a basic principle, I think any breeder or producer want a sustainable, want to sustain, especially economic sustainability is important. So like I've mentioned, carriers of animals, and then also changing mating programs that relied on the average relationship matrix, changing those with the actual genomic or the information that comes out of the genomics in terms of real genetic relationships. So yes, there's always the notion not to inbreed if possible, or to keep the rate of inbreeding as low as possible. So to maybe not the way you coined your question, but in essence, that is the objective, to do just that, so that you don't run into bottlenecks and you outcross lines, to assist you to retain at least some margin of genetic diversity. Maybe something else that's very important is the fact that how breeding objectives has been formulated lately, where a lot of different traits with the genetic relationship amongst those traits being included assist to a large extent to just stop you from mating very close-knit families, as we've seen in the old days in milk, for example, where milk production was pursued, but longevity and all the fitness traits went down the drain. And I think there's been a realization to include more traits in these objectives.
If I could maybe add to that question, Dr. van der Westhuizen, you also now refer to this balanced approach of selection, and then doing that together with genomic selection. We've implemented that approach now in South Africa for a while. So do you have an indication, and I know this is a difficult question, but of the economic value that this has produced, the benefits in a monetary way that it has given our farmers in, let's stick to the beef cattle industry?
Yes, I think that's a very, very important point. Different countries call their selection index by different names. We call it, in the beef cattle, we call it the cow value. And that's basically a monetary value per hectare or per unit of land. And that would then be built on the building stones or the building blocks of ease of calving, female fertility, longevity or effective herd life, maternal ability, pre-wean growth, and also maintenance requirements of your animals. So by combining those building blocks and the underlying traits into a single value, that is then basically a monetary value. But you can have two animals with exactly the same monetary value genetically, but the one gets there through fertility and the other one through, let's say, good maternal ability. So, and that assists also not to suck up all the variation in selection for those single traits that underlie, those selection indices.
Thank you so much. Any other questions from the floor? I think we are done, and I think we are out of time. Thank you so much, Dr. van der Westhuizen, for this interesting talk.
Thank you very much.
We are then now moving on to our two shorter presentations. It's the young scientist speakers, and the first one is Dr. Nesengani, who's going to talk about the draft reference genome of the South African Nguni sheep as an output from the AfricaBP pilot project.
Seems like we still have a bit of glitch. Well, in the meantime, good afternoon, everyone. My name is Lucky. I'm always lucky every time, but this time it doesn't look like it. Should be fine. Okay, I'm told we should be good to go. All right. Okay, maybe I need to change this name. Display, let's display. So automatically it should just. Is it possible I give you them the USB you project from your screen? Okay, can you bring your laptop? Okay, we should be good. No, it should work. Okay. Okay, apologies once again. I know we're still a couple of minutes from your lunch, but now we should start. Okay, once again, good afternoon. My name is Lucky. I'm here to give a talk on the output of one of the project that we've been working on, specifically on the draft reference genome of South African Goonie sheep. It's an output from one of the projects under AfricaBP. I'm not gonna emphasize a lot on the AfricaBP. I think Prof. Anne gave a good talk on the outlining all the goals and the purpose of AfricaBP. I'm audible, right? Okay. I just love my voice. It's very fresh. I thought maybe you can't hear me. So yeah, like I said, the AfricaBP, most of the purpose and the objective have been outlined in the morning. So I'm just gonna focus on the work or rather the research that we've been doing. So basically, why do we do the genome sequencing, or rather, why do we need the genome references? They are actually the essential resource for the future of genetic breeding to better understand the organisms, the entire genome architecture. We need to develop high quality reference genomes. But if we look around the world, we're still having quite of some of challenges where some of the species don't have high good quality references. This impact on the research output out there, as well as this is basically the issues that most of the references that are out there, they were generated using long short reads. I mean, short reads, which is not rather an ancient technology, but it had its own challenges, such as accounting for some, when you're doing the reference genomes, you couldn't encounter for some of when the genome is big, it has a lot of repetitive elements. You talk about your transposable elements, such as the tandem repeats. These short read sequences couldn't account for that. But fortunately enough, in the wake of technology advancement, we're now in the position to generate long read sequences, which we are fortunate enough to, we can account for those challenges. But what do we know so far with this technology that is presented before us? We have seen with the publications of the human genome, which is, it was a complete genome of telomere to telomere, which it has, we can say it's a near error-free genome. It's got quite a good quality. And this also corrected some of the challenges that I've already mentioned, the issue of the repetitive elements. So we see that it's a good approach actually. And but however, we still have those ones that were generated before that have challenges of that were not, they have quite long gap that we need to rework on them in order to account for those repetitive elements. Looking at it from the African context or perspective, rather than majority of the African species, we know that they don't have reference genomes. Even those, we didn't even, majority of them don't even have the short read reference genomes. And that for that reason, we know that I'm glad that the previous speakers have indicated the importance of our indigenous species, specifically in Africa, that they are quite of significance, especially when you talk about the food security. They have quite good genes for adaptability and resistance to some of the challenges that we're facing in the continent. Even Professor Yapi just previously mentioned there's the importance of guarding against the dilution of the indigenous species. So in order to advance their production, we need to understand, to study their genetic architecture, to understand the genetic makeup of the organisms in order to manipulate the organism to improve the production traits as well as the adaptability traits. After evaluating this challenge that we've seen that the majority of the African species do not have these reference genomes, we have set up a Nelson Mandela Genomes Project through Mapodi Labs in collaboration with the AfricaBP. The general aim of this was to develop or produce high quality reference genomes sequences for African indigenous species. One step at a time, we will reach there in the telomere to telomere complete reference genome. God be with us. So, like I said, in this talk, I will specifically focus on the Nguni sheep. What do we know about the Nguni sheep? The Nguni sheep, we know that they are indigenous to the Southern Africa region, and they also have the good part with them. They are well adapted to tropical conditions as well as most of the external and internal parasites, but as well as resistance to some of the problematic diseases, such as the tick-borne diseases. But however, we have currently seen that this precious species, precious breed, it's facing a severe population decline due to crossbreeding with exotic breeds. I mean, if the farmer is always looking to increase productivity and if they see the other breed is doing well, the solution is always to crossbreed. But in the meantime, we're also sacrificing the good traits that we're not paying attention to, which we're talking about the traits of adaptability and all that. So the genomic information that we need to develop a better genomic information or genomic baseline that will be used to develop breeding programs. These programs should specifically be used to, or rather will be beneficial for use in the programs in order to preserve this precious genetics. So what did we do here? in order to develop some of these imported genetic tools, we took a trip not far from here. It's on the on the northern side of Limpopo. Those ones who don't know, the Mara Research Station is located in the far north of Limpopo, not far from Makado area. We went to sample the Nguni sheep with the help of the vet. We collected the blood samples where we extracted the high molecular weight DNA that was subjected to be sequenced at the PacBio with the PacBio sequel and on the Nova 6000. After the sequencing, we obtained the total Hi-Fi output data for Nguni sheep was about 99 gigabyte, which was at a coverage of 31x, while the output for OmniC was 300 million reads pairs. So this is just to show the genome scope profile of the Nguni sheep. It has a total, the genome size of about 2.97 gigabyte, with the homozygosity of 99.8% and the heterozygosity of 0.24. You can see that the peak of the average coverage was about 32, 31, somewhere there. So the assemble statistics, how does it look compared to the assemble statistics for the Nguni sheep? How does it look with the other output or published genomes out there? We have seen that the genome size actually, it's not bit comparable with the other genomes that have been published, as well as if we can look at the scaffold N50, partially not bad, good. I know the VGP recommends somewhere that should be the scaffold N50 should be around 100 megabyte or somewhere between there, as well as the Quantic N50 should be at least 10 megabyte. Excuse my laugh. So we further went to check at the Basco output and compare them with the others that are out there. I'm not sure if it's very visible, but I have, like I said, I have a very good voice. You can listen to my voice. I can read the numbers. So the Basco output for the Nguni sheep, it's actually comparable with the other reported genomes of the Nguni out there. We obtained in total 96% of the Bascos, which is comparable with the Remelot sheep that got approximately 96.8%, I think. And the others were a bit lower than what we got. The others, there was the other one that got 93% of the Bascos. So I think the quality, oh, and also I forgot to mention that this was done by Africans in Africa, which is something that we take pride that is compatible with the work that is out there. So everything from sampling, sequencing, and analyzing. I need to take pride. So this is the snake plot just to show the other assemble statistics, showing the long read, the long scaffold, as well as the log10 scaffold, and also the Hi-C map there. I did put it there, but it's not yet completely free of the misassemblies. It's something that I'm still working on, the manual curation. We know that it's time consuming and labor intensive, but we are getting there. I'm focusing on it. It's one of the important steps actually in getting a near gap free genome references of high good quality. So yeah, we will get there. And the next step from here, it will be to work after the manual curation, we will take it to the annotation part. This is also, like I said, I'm always liking to take pride of the work that we do. I was asked to talk about the Nguni sheep, but I also wanted to highlight some of the work that we're doing. This is specifically on the Bonsmara and Nguni cattle. We have also completed the work there. This is how the assembly statistics look. Okay. This is how the assembly statistics look, and it's actually looking quite better, compatible as well with the other already published genomes. So what is it that we take? I mean, we're doing this work, so we know we're still going to look at the functional part and also do the post-assembly analysis and look at what we take good from it. So just to emphasize on the value proposition of this work, the telomere to telomere genome completeness will provide opportunity to explore the genetic architecture of indigenous species. In this way, we will be at the unique position to better understand and to also to plan on the programs that will be developed, especially the breeding programs, to improve some of the economic traits. We talk about the heat stress and also the production traits, and also for conservation purposes. I mean, it was emphasized quite a lot. Even Dr. Isidore did mention the challenges of losing our indigenous species earlier on his talk. So these are the opportunities that we need to explore or tap into this technology in order to preserve our precious genomes. So this work will definitely ensure breeds long-term sustainability as well as economic viability and contribute to food security. So what is the take-home message from this talk? It's having a high-quality reference genome that is accessible to researchers is essential as a genomic tool to establish efficient breeding programs. This will definitely lead to one of most of the things that I already mentioned, that we need this in order to preserve and also to improve the indigenous species. Thank you.
Thank you so much, Lucky. Well, you told us to call you Lucky, so now you can't be Doctor anymore, okay?
No, I'm Lucky.
Any questions for Lucky? Okay, otherwise I have one for you.
Well, I'll just make a comment for the scaffold N50. The minimum we set for the VGP was 10 million base pairs. So 10 megabytes, not 100. But if you get 100, that's really great. Yeah. And it's almost near telomere for vertebrates.
Thank you.
Okay. There was another question.
Thank you very much, Lucky, for the wonderful presentation. Mine is just a quick one. How much did it cost you to sequence?
Okay, that's an interesting one. I think for one complete genome, when you include the Omni-C, the Hi-Fi, she was somewhere around 230. Yeah, 200. Yeah, I think Promopur can correct me if I'm not mistaken.
345, kind of.
Okay.
So that's if you now cover the cost for long reads using the sequel 2 then. And then you do your Omni-C using the Illumina Nova 6000. But if you do it now with the PEG by Revio, it costs you about 75,000 rand just to get your 30x coverage. Then because now there's also NovaSeq new one 6000X plus or X, that now it also cuts your cost down. So more likely to spend about 125,000 per genome to be able to have both the Omni-C and the long reads.
Okay, yeah, there goes my banker.
Yep.
I don't know the banking things.
There was Prof. Berger, another question.
Thank you very much. I'm just curious, you got quite good BUSCO scores, but you didn't mention, do you do a lot of RNA-seq as well to be able to help with the annotation, or how are you able to actually annotate the genome?
We haven't annotated yet, and we don't have the RNA-seq for the ones that we have now, but we are going to make use of the already available sequences from the reference genome for annotation.
So you're not going to do RNA-seq from that particular Nguni sheep sample?
Yeah, the plan is for the the plan is for the next ones. We will definitely do the RNA-seq, but these ones we can do the annotation using the reference genome that is available in the NCBI. But that's a good one that we really need to do both the RNA-seq as well.
Okay, thank you.
Very quickly, quick.
Maybe if you can say.
Yeah.
Okay. Yes, we are still waiting. Dr. Smith is just quickly doing something. So, Lucky, I have another question for you because we have to keep the
Oh my
audience
God,
busy.
I thought I was done.
Now, I was just wondering, you said all of this will lead to the improvement of production traits and reproduction traits, etc., meat quality. Do we have phenotypic data for Nguni sheep on those types of production traits?
Well, I think it goes back to the earlier discussion on the better developing the recordings of our species. So we also need to work on that. And especially on I liked the issue where they emphasize on how do we better standardize the recordings that we're doing. So it's something that we need to think around on how do we better develop these recordings of not only for the purpose of population reference population, but also the recording of all the animals that we have. How do we better improve if we don't know much about it?
Yes.
But we definitely need to start somewhere.
Absolutely, I agree. We need to start somewhere. Okay, I think we are almost there. Our last presentation for this morning session, which is now afternoon session, is another young scientist speaker, and it's again an output from the AfricaBP pilot project. This one is on the draft reference genome of the South African Colebrook, and it will be done by Dr. Smith. Thank you.
It's requiring me to sign in. Okay. Just wait for it to be loaded or displayed. Okay, thanks everybody for your attention. My name is Ray Smith. I'm also part of Mapoli's group, and I'm also here just to showcase some of the work that has been going on in our efforts to contribute to the Africa BioGenome Project. So some of the work that we've had is we've received samples that we are slowly developing our skills and expertise on just engaging with the genomes that we've been provided with, and hopefully you'll get an interesting story of how that is taking place. So the Coolbrook pig is an indigenous breed to South Africa. Some people had an initial idea that it was related to the Chinese breeds based on its phenotypic characteristics. But other facts about it is that it's an excellent forager. It's resistant to South African the diseases within the South African climate. It's also, it's not a very productive pig relative to the commercial breeds, but it's very robust in terms of its production capability within more smallholder environments. So something that has also been noticed is that there is a potential for introducing hybrid vigor or heterosis, leading to the production value of 90 to 100 days to market. So some of the previous work that has been going on with this breed is some general population it's investigations where initially microsatellites was investigating microsatellite alleles to see how genetically distinct the Coolbrook population is relative to commercial breeds as well as other indigenous breeds. So that work has slowly been going on to show how distinct the population is. And relative to the South African breeds, there is, what was reported was it's actually more inbred relative to the other breeds, but there is a definite distinction in terms of its genetic diversity. So to put the question of whether it's related to the Chinese breeds to bed, public studies have continued to show that the indigenous breeds of South Africa are not related. So that was just an interesting recent development. So the aim of my study was also just to work on developing a high quality genome of the Coolbrook pig. And what I'll be showing you now is my progress on that endeavor. So I think what Lucky has given a very good forward to the work that I've been doing. So initially, ethics has been applied for through through our local government's departments, Section 20, and the Research Ethics Committee. The sample was collected also through the Amara Research Station. So the general preparation leading up to this would be the DNA extraction method, and then preparing the libraries. So what Lucky mentioned as well, so using the Smart Bio kit and the Dovetail OmniC kits, which are run on the PacBio SQL 2E and the Nova 6000 respectively. So just a small peek into the workflow that we've been using to train ourselves on, as well as to perfect the technique of generating these near complete genomes. So we run about 90 gigabases of Hi-Fi or gigabytes of Hi-Fi data that was run on the SQL, as well as an additional 90 bytes of OmniC data. this goes through the workflow that is produced and curated by the Vertebrate Genome Project, where we go through a series of steps, moving through cut, adapt, generating a Mercury database, and then running it at initially generating a KMA profile using Genoscope 2. And then in addition to that, then assembling the contigs using Hi-Fiism. And throughout this process, we are busy trying to assess the validity of the contigs that, or the sequences that are generated or assembled. So we use the BUSCO, the GFA stats, and Mercury to assess that along the way. As we move along the process, once we've got our assembled contigs, then we continue with the process by assembling using BWA MEM2, as well as extracting the mito, the mito genome using the program PacBio Hi-Fi. And then the process for assembling the scaffolds is continued using the yet another Hi-Fi sequencer. And then these results are then visualized using pretext. And in addition to that, generating the snail plots and decontaminating using programs like Kraken 2. An additional inquiry that I had was also just to see how it compares to the reference genome that is currently available. It was produced in 2022 by Amanda War and colleagues, and I just wanted to see how this genome that we had, which was at 31x coverage, how it compares to their 61x coverage genome. So then the results that I've obtained for my efforts was that we were able to generate a KMA profile that yielded a 2.6 base pair or 2.6 megabase pair estimated genome size, and it was at a heterozygosity of about 99.7, with a reasonable the sequence depth correlated to what we expected, which was 38 times. And throughout the process, we were checking the BUSCO scores against the lineage of Seta Areodactyla, and we got about 95% similarity with with about 13,000 sequences or 13,000 profiles. So the pretext map that was generated did show a bit of trans interactions, or yeah, between the scaffolds, but overall it was quite a clean sequence, a genome that was generated at this level. So I went on to compare the genome that, or the scaffolds that was generated through this workflow with the current reference genome, and this dot plot over here represents what was generated from a program called Degenez, and it's just comparing the two genomes together, where seeing a diagonal line, a perfect diagonal line would indicate that there's 100% similarity. There is some interactions here that will require some manual curation, as Lucky was also saying. So those are some of the steps that I'll still be working on in future. But in terms of the assembly itself, the N50 of the scaffolds, I think it's slightly above 10 million. It's about 280 million. So I think it's a quite a good result for what we would hope, especially with the level of coverage that we had. And what I also saw, we've also extracted the mito genome as well from the sample that we've collected, and this is just a representation of what we've of it being annotated using the currently available reference data. So moving on to the discussion, I would just like to say the genome that was generated in 2022 is quite high quality, and they've applied also, I think it was SQL2E as well that they've used as well, and went to a depth of 65x, as well as did some hybridization against the physical chromosomes to confirm the integrity of their genome. And from what we've produced in using just the VGP workflow and just a bit, not too much tweaking, we've actually generated quite a high quality genome sequence. So there was some unallocated scaffolds, but I also suspect that some of the other elements, such as the mitogenome, which is not displayed with the other in the dot plot, was also there, and some other elements as well. We did try to decontaminate as well, but that didn't yield any significant differences when it comes to visualizing the genome. So I think just to conclude my talk, hopefully I was nice and quick. We've generated a high quality genome that is relatively comparable to what is currently available in terms of the reference sequence. But there's still a bit more work that needs to be done in terms of the curation of the genome. But with this workflow alone, we are able to generate successfully high quality genomes and do it at a very rapid rate, which is in line with what we're trying to achieve through the AfricaBP platform. So just some future work that I would like to do moving forward is also just continue with the manual curation, and then just investigate more closely the synteny between the reference genome and the current assembly, as well as also, for my personal interest moving forward, study the immunogenic differences between our indigenous livestock as well as the commercial breeds. And with that, I would just like to acknowledge the following people and thank you for your attention.
Thank you, Dr. Smith. Okay, last speaker must get the most difficult questions. So, who's going to go first? We can't let him off this easy. You go, yes.
Yeah, I know there with other genomes, right? In the same sequencing centers, there was challenges with the Hi-C. And here, this Hi-C worked, OmniC that is, worked well. And I'm wondering, do you think that's purely a sample, in a quality difference, or something else?
So in some of the other samples that we've had, there is some challenges as well. So it's a bit early for, I think, when we compare each of the genomes that our group is working on, we'll be able to more accurately say whether it's the sample quality. But I think our partners that we've working with, they've really emphasized the sample collection step. So I think in terms of sample quality, we are there's definitely good procedures in place for ensuring that the sample that we collect retains its fidelity towards the point of sequencing. Mm-hmm. So there could also be internal challenges as well in terms of the genome itself. So we'll continue investigating that as we develop or as we produce more genomes within our group as well. So yeah.
All right.
Olivia, did you want to add something?
To ask Ray, because I can't remember now if in this one that he presented, did you run it with the upgraded?
So the results that I'm presenting here, so just for context on that question, the VGP group are constantly upgrading and curating their workflow. So the version that I've used is not the latest version, but irrespective of that, the data that was generated still yielded very high quality results. So what I'll do in future steps is also just to rerun it, seeing where the differences with the programs used in the workflow vary, and see how that affects the assembly. So, thank you.
Okay. Are we done? One more question?
To finish. So firstly, I realize you've been concentrated on the mechanics of generating your genome. And for that work, you should be congratulated. It's excellent. For that to make it hard question, though, I'm more interested in some of the selection pressures that the smallholders have applied on this breed and how you're going to bridge that reality in the field across a large number of small holdings to the information that you're generating now.
Yeah. So
you set the challenge.
Yeah, I think it's a real challenge. So even in my previous experience working with smallholder farmers, they are very under-resourced and they have got limited access to sort of the resources in terms of educational resources to be able to make the kinds of decisions in terms of trying to improve their own breeds. So it's a big challenge, and I think working hand in hand with trying to promote also better breeding strategies as well. So the work that might come out of this, especially in terms of my interest in being proliferating the adaptive traits, having a good education plan, or meeting with the smallholders by trying to make use of various online platforms to support them in spreading that information, because it is a big challenge. Yeah.
Thank you. And thank you for the question. Olivia, one sentence. Olivia, I think if you put 1 more minute between these people and their lunch, there's going to be a riot. So thank you, everybody. And I just want to thank our speakers again, everybody that contributed to this session. Thank you very much. Enjoy your lunch. And for everybody that just walked in, no, we are not in the afternoon session yet. We are first going to have lunch, and then it will be the afternoon session. Thank you very much. Yes.
What time will we start again, please?
Olivia, what time do you start? Greetings.
or new run configurations. Maximize batching and scale for applications like exome, single cell, and spatial sequencing on the market-leading platform. Do more with P4 on the NextSeq 2000.
Extreme importance of the private sector, especially the animal geneticists, to be part of such an important program, such as the African BioGenome Project. I was asked to talk about the application of animal breeding and genetics, with a special emphasis on genomics in the African bioeconomy. And I think we've seen strides being taken lately also on our continent in the development of or using genomics in the bioeconomy, especially in my case, and in livestock or production animals. To kick off, I think one should start at the beginning and say, what are we trying to achieve? And in my mind, what we are trying to achieve is to unravel the phenotypic differences amongst the animals.
Mic testing, 1, 1, 2, 1, 1. 1, 2, 1, 2. 1, 2, 1, 1, 1. 1, 2, 1. Hello, hello. 1, 2, 1, 1. 1, 2. 1, 1, 1. Is it sharp? Or hello, hello, hello. No feed. Hello. 1, 2, 1, 2, 1, 1. 1, 2. You don't hear anything. 1, 2. 1, 2. 1, 2. 1, 2. 1, 2. Because they are here. And then you want to push 8 feet. That means they must come close.
chemistry, nanoscience, and other disciplines. MGI is one of few companies in the world that have the ability to develop and mass-produce low, medium, and high-throughput clinical gene sequencers from GB to TB. Besides gene sequencing, MGI has also developed a series of core technologies, including library preparation and automated sample processing. Expanding into new business, MGI has developed multiple innovative life science tools and technology, including portable single-cell systems, remote robotic ultrasound systems, BIT products, and low-temperature storage products, with a strong focus on the upstream of the industry. MGI operates in more than 100 countries and regions, serving more than 2,800 customers to date, empowering the downstream customers in reproductive health, precision cancer diagnosis and treatment, infectious diseases, food security, agriculture, forestry, fishing, and consumer genomics. Since the establishment, MGI is driven to disrupt the status quo and accelerate the industry innovation. MGI aims to build a suite of life science core tools to address the fundamental questions, push the limits of science and technology, and lead the innovation in the era of life.
Hey there, you're such a tiny thing, but you brought great to the world. Your story became much bigger than we ever dreamed. People lovingly call you Epi, and you changed how we work in the lab every day, more than once. With every little drop that you've held, great inventions could be made. You like it cold. You like it hot. You like it shaky. You're colorful and convertible, and your family grew bigger. There's nothing that can harm you. And that's good, because you preserve for us what we adore most, what's everything to us, the sample.
The new P4 flow cell. Experience more reads, greater accuracy, and the most cost-efficient runs on NextSeq 2000. Expand the utility of your benchtop system with 4 new run configurations. Maximize batching at scale. For applications like exome, single cell, and spatial sequencing on the market-leading platform. Do more with P4 on the NextSeq 2000.
Quang tên hỏi quang thuộc cả nó có nè. Quang tên hỏi thuộc cả này. Quang tên hỏi thuộc cả này có nó. Quá thì bệnh. Quá thì là hụi. Cũng thay chế mà phần nát. Mà lúc tê cài lắng anh ấy chế. Ai chào hụi lắng bọn ấy chế. Anh ấy chế. Léo bọn ấy chế.
It's projected that the world's population will grow another 2 billion people in the next 20 or 30 years. These fish look in really good condition. The key thing here for farmers is to be able to produce enough fish to feed the population in his area, but also globally. My name's John Benzie, and I'm the Acting Director of Aquatic Foods Biosciences for World Fish, based here in Penang, Malaysia. Fish are highly nutritious and in many parts of the world are the key components to people's nutritional security. It's important to produce more of these foods. One, because they're nutritious and they've been fed today, but secondly, because there's a huge demand for them that is not currently met. In this hatchery, we maintain the genetically improved farm tilapia strain known as GIF. This fish now grows twice as fast as unimproved tilapia.
Let's try. Yes. Yep. Okay, so let's go.
When the fish are large enough,
Slowly.
we take a small piece of the fin. Doesn't harm the fish at all. And we take that sample to provide to our research partners in the UK, Errolham Institute.
Hey guys.
Hey, Taryn.
Where we come in as the Earlham Institute,
You've got lots of night.
what we do is we look to sequence the genomes of these fish. It's quite small fish, and then annotate the functional and non-coding aspects of the genome to then better inform how they could then improve these fish further.
We could utilize the ability of the Nile tilapia to grow particularly well and fast.
I'm Kiran Gandhi, I'm head of genomics pipelines at the Earlham Institute in Norwich, England. The issue that we're trying to tackle with this collaboration with WorldFish is to try and improve agriculture in Africa and Asia of tilapia fish. To do this, we use state-of-the-art technologies to look at the genome as a whole, and in some cases, very specific areas of the genome that might be of interest and linked to these traits that the farmers are interested in. So the benefits of the NovoSeq for this type of projects are twofold. The first one is reducing the cost. The second benefit to us is the speed.
What's really important there is that allows us to apply multi-omic approaches, which gives us better power to translate some of our findings to deal with my mission of improving food sustainability, but then also Earlham Institute's mission to also protect life on Earth.
This is my passion, my scientific passion really. So being able to connect the technology that we've set up in this lab to the life of farmers and the livelihood of farmers and helping them thousands of miles away with the work we're doing here in the lab is fantastic.
It's going to be important to produce not only organisms that grow faster and produce food more quickly, but also do so with less environmental impact. Feels like a worthwhile thing to do, and it's satisfying at the end of the day to think that you've made at least some small contribution to that process.
For over a decade, Illumina's solutions have enabled researchers and public health scientists to make groundbreaking discoveries and protect our communities from infectious diseases. From enabling the adoption of whole genome sequencing to aid investigations of foodborne outbreaks, to more recently helping public health officials around the world track the transmission of SARS-CoV-2, Illumina has been dedicated to those studying the smallest and deadliest threats to human health. Today, we are proud to introduce two new library prep solutions built on the simple and affordable COVID-seq workflow that will help build global genomic surveillance capabilities and power impactful microbiology research. Illumina Microbial Amplicon Prep, or IMAP, is a unique library prep solution allowing researchers to use lab-designed or commercially available primers for targeted sequencing of viruses and select markers from larger microbial species. Ideal for One Health applications, the kit provides all the reagents needed to go from extracted nucleic acid to sequencing-ready libraries. Illumina Microbial Amplicon Prep Influenza A/B, or IMAP-Flu, is optimized to provide extensive sequence coverage across both influenza A and B viruses, compatible with RNA inputs extracted from cultures or nasal swabs and washes. IMAP-Flu delivers exceptional data quality, flexibility, and scalability for public health surveillance and high-impact influenza virus research. Both solutions seamlessly integrate with the Dragon targeted microbial app. This powerful app serves to simplify data analysis, making it easy to perform consensus genome generation, variant identification, and variant interpretation. IMAP and IMAP-Flu, two exciting new additions to Illumina's portfolio of solutions for public health surveillance and microbiology research. Because in the genome era, infectious diseases don't have to be a disaster.
Human beings have been using astronomical tools to adventure into the infinite universe, to explore the infinitesimal by making use of life science tools and technology. Founded in 2016, headquartered in Shenzhen, China, MGI is committed to building core tools and technology to lead life science through intelligent innovation. MGI has established 7 international R&D and production centers and is mainly involved in 3 key business segments, including genetic sequencers, laboratory automation, and new business. Continuing heavy investment in R&D, MGI has developed a full range of patented technologies, including DNB-seq sequencing technology, patent array technology, and sequencer optoelectromechanical system technology. Gene sequencing has a very high barrier to entry as it involves multiple fields, including optics, mechanics, electronics, fluid dynamics, molecular biology, chemistry, nanoscience, and other disciplines. MGI is one of few companies in the world that have the ability to develop and mass-produce low, medium, and high-throughput clinical gene sequencers from GB to TB. Besides gene sequencing, MGI has also developed a series of core technologies, including library preparation and automated sample processing. Expanding into new business, MGI has developed multiple innovative life science tools and technology, including portable single-cell systems, remote robotic ultrasound systems, BIT products, and low-temperature storage products, with a strong focus on the upstream of the industry. MGI operates in more than 100 countries and regions, serving more than 2,800 customers to date, empowering the downstream customers in reproductive health, precision cancer diagnosis and treatment, infectious diseases, food security, agriculture, forestry, fishing, and consumer genomics. Since the establishment, MGI is driven to disrupt the status quo and accelerate the industry innovation. MGI aims to build a suite of life science core tools to address the fundamental questions, push the limits of science and technology, and lead the innovation in the era of life.
Hey there, you're such a tiny thing, but you brought great to the world. Your story became much bigger than we ever dreamed. People lovingly call you Epi, and you changed how we work in the lab every day, more than once. With every little drop that you've held, great inventions could be made. You like it cold. You like it hot. You like it shaky. You were colorful and convertible, and your family grew bigger. There's nothing that can harm you. And that's good, because you preserve for us what we adore most, what's everything to us, the sample.
Okay, good afternoon, everyone. All right, welcome back to the afternoon session. We're going to get Prof. Maharaj's presentation up just now. My name is Prof. Dave Berger. I'm from the University of Pretoria, just up the road. I'm a plant molecular biologist, plant geneticist, genomics kind of person. So it's quite exciting to be here. I was interested to listen to the animal talks earlier today. And so we're going to have a keynote from Professor Vinesh Maharaj, who's our Deputy Dean for Research in the Natural and Agricultural Sciences faculty. And we're going to have that presentation, a live presentation, and then there's going to be 3 virtual presentations after that. So Professor Maharaj, you'll hear he's a chemist, but he's worked a lot on indigenous medicinal plants, endemic medicinal plants, and the interesting things that come from that. So Professor Maharaj, it's a pleasure to welcome you here, and I'll hand over to you. Thank you.
Hey, good afternoon, colleagues. Thank you, Dave. And thank you to everyone else and the organizers as well for giving me this opportunity. So the title you see in your booklet is somewhat different to what I have up here. After I realized what this workshop is more about, I've decided to change it to be more in line with your objectives for today. So really what I want to talk about is more looking at natural products in terms of its chemical diversity. So it's a bit different from what you've probably experienced so far, looking at your genetic diversity. So I'm going to be talking a bit more about chemical diversity from our genetic resources. And what our overall aim and objective is how do we create a natural product chemical library? You've heard a lot about databases on genetic libraries, and our objective was to try and create a chemical diverse library, mainly for the purposes of pharmaceutical type-based research. So as Dave says, I'm the Deputy Dean of Research, but I'm also the director of a center at the University of Pretoria called the Biodiscovery Center. And whenever I have spare time, I manage to get some research done in the center. Okay, so we all know about our great biodiversity and our mega diverse country, or third highest in the world. But of course, if you want to utilize this diversity, especially our plant biodiversity for medicinal purposes, particularly if you want to use it for drug discovery purposes, And we know if we are successful in this, the contribution to bioeconomy can be quite enormous, especially if you're actually able to launch any pharmaceutical drugs based on our biodiversity. However, with our huge biodiversity, we always ask our question, so where do we start? And the approaches can be almost three-pronged type of approach. You have the chemotaxonomical approach, is basically saying, if we know a certain class of plants that contain certain classes of chemical compounds that are biologically active against a certain disease, you can target those plants and work for that disease. Alternatively, and in South Africa, you know, and even in Africa as well, that there's a rich heritage of indigenous knowledge systems. So many of our plants are recorded to be used for medicinal purposes. And that's actually quite useful because that actually narrows down your search, and you can reduce your search to a few plants and focus on a specific disease. So that's often called the ethnobotanical approach. And then, of course, there's the random approach, and you can give yourself a target and say, well, let's just collect everything and test everything. That's a huge number to go through. If you look at the South African biodiversity of 26,000 plants. So it's quite a challenge to be able to collect all of this, and then a bigger challenge to be able to put this into a sort of a machine so that you can actually get them analyzed and tested. So those are the three approaches that one can have in trying to discover drugs. And of course, all of them are underpinned by having very good biological systems to evaluate these for different diseases. Right, so most of you, or many of you, may be familiar with this approach. If you're in drug discovery, you actually want to find the so-called golden molecule that exists in these plants. And we are very familiar with the process of bioassay guided fractionation, which typically takes a plant, extract with some organic solvents, do some bioassays, and then go to the tedious process of separation these thousands of chemical compounds that exist in this complex matrices, and ultimately, through an iterative process, come up with a compound which may or may not be biologically active. And by the time you get to the end of that, you've realized that this is like 1-2 years down the line. You identified a compound that is well published or known to be active against the disease. It's an artifact. You have many artifacts in plants. And there's various other reasons. And you also then spend using lots of biological material. So you need a lot of plant material to get to a few milligrams of a compound that you can develop as a drug. So there's many challenges about following this classical approach. And as a result of it, there are new paradigms now as to how you're going about doing that. Especially in drug discovery, it's a known fact that about 1 in every 10,000 compounds that enter the drug discovery pipeline, only one of that actually makes an approved drug approved by the FDA. So the dropout rate is very high, is very costly, and can take a long time, up to about 10 to 15 years, just to get one on the market. All right, so how do we improve this? There's different ways of trying to increase your chances of success, and that's one is to increase the number of compounds. So you can go from 10,000 to 20,000, but then it's going to cost you twice as much. Okay, the other way is to increase the quality of what you put inside. Instead of using just 10,000 any synthetic compounds, you can use natural products. And we know that natural compounds actually have a very high, what we call hit rate, because of their long history of uses, and they have a good chance of success. So focus the attention on natural compounds. And more recently, the approach is now to use artificial intelligence and machine learning to actually put millions of chemical compounds just through an AI model that will narrow down your search to 1 or 2 that can go into the development stage. So those are really, if you're talking drug discovery, how difficult it is to work on basically 1 or 2 plants at a time. So as a result of this, those involved in drug discovery, natural product research, have resorted to creating libraries. And some of the most well-known natural product libraries around the world is one at Griffith University in Australia. That was created there of about 200,000 samples from plants and marine organisms. There's one in Singapore. There's a few others as well, but I'm just talking about the major ones. There's about 150,000 in Singapore. but perhaps the largest one in the world is known as the National Cancer Institute, where they have 2 million natural product samples that they put into high throughput screening for anti-cancer research. And of course, if you know, this is the National Cancer Institute had come up with quite a few drug leads, well-known Taxol, which is actually on the market for cancer. All right, so we gave ourselves our target. Let's try and do something similar in South Africa. So our goal was to create a diverse library of natural products, extracts, compounds, and fractions that we could collect from plants, microorganisms, and marine organisms, put that into extracts, and then purify these through high throughput purification, and ultimately put them into storage. So we can then actually use this for high throughput screening campaigns against different diseases. We started off by a collection which spanned around 4 years. This was supported by us with Sanbi, who did all the taxonomy of all the plants that we collected. We got to a point where we managed to collect at least 11,000 plants, and those were all dried and stored in sort of a repository that we had. We had a very unique sample numbering system collected from most of the biomes on the country, from the Western Cape right up to Northern KZN. And of course, we had to create a database for the management of the samples, because the samples were all converted into being curated, plus also converted into extracts. So we had a good collection of plant samples collected throughout the country, and I believe this is probably one of the largest on the continent, and actually having a repository of 11,000 plant specimens. Right, the next we actually said we need to convert this genetic diversity, which is your plants, into a chemical diversity, so we can actually do high throughput biological screening for different diseases. I was fortunate enough to get funding from the Department of Science and Innovation to the tune of about 50 million rand, and we were able to we started setting up the platform to be able to do that. So it basically takes the plant, and most of it is actually automated. So we have a system for extracting the plants using organic solvents, and then moving that into what we call a purification step. So we have liquid handlers that automatically separate each extract into different what you call fractions. These are semi-pure, and finally then take into a liquid handler that will actually format all of this into what I call a microtiter plate, actually removable tubes, and eventually put it into a robotic freezer stored at 5 milligrams per million DMSO. And that's what we call a mother plate, stored in the robotic freezer at minus 20 degrees Celsius, under nitrogen as well. So it's all dry as well. And so that's ready for screening. We managed to get about 5,000 to date. That's an ongoing process. When I talk to my students by next week, they'll have another couple hundred that they keep on adding on a weekly basis. So that's typically what a plate looks like. You have a plant with these different extracts and all the different fractions for each plant in a column. So that's how these samples actually stored. So whenever we go into a screening campaign for a disease, we would take a copy from the libraries, from the freezers. So we'll make a copy, and through the liquid handler again, we'll then transfer a portion into a copy into another microtiter plate, and we'll seal that, and we'll send it to whoever needs to do a biological assay. And typically they would do a high throughput screening. And of course, whoever is doing high throughput screening needs to have liquid handlers as well. Everything is barcoded as well, so nobody knows what sample they're getting either, but just go into high throughput screening. But once you get a hit, the next challenge is to say, well, this plant X gives you very good biological activity against disease. A certain disease. You then need to identify what is the chemical constituents in there. We typically have a UPLC QTOF, mass spectrometry, time of flight, which we typically use to actually get a tentative identification of what's in these wells. And because they are purified, it's actually less complex. And ultimately, we have what you call mass-directed HPLC. So once you know the mass, we are able then to direct your purification, focusing on that specific chemical compound. And ultimately, through NMR and XRD, we're able to tell you what's the chemical structure. And because we're working with micro quantities here, we have what you call an NMR, which has a cold probe. So it's typically in the region of about 800 megahertz. It's a 500 megahertz with a cold probe. So the power for in terms of the magnet, it's equivalent to 800 megahertz NMR. And we typically can get an NMR for those of you a year on about 200 micrograms of samples. So that's quite a small quantity to get data. All right, so I just want to move into some of the case studies that we've done and the success we've had using this approach. So one of them has been actually testing for antiplasmodial activity. That's the malaria parasite. We've screened 256,000 samples. Unfortunately, this could be done at the University of Pretoria through the Institute for Malaria Control. And so we screened lots of plants, lots of samples, and then we identified those that are hits. And typically a plate map would look something like that. The red looking at those that actually look very good in terms of activity. So those are the ones that we would then focus on actually. Specifically, there were 3 plants that looked very good. And by the way, Artemisia afra, we've tested for every disease. I'm sure you know Artemisia afra seems to light up in every assay, seems to work for everything. So we ignored that one. And we moved on to the others, was aloe and torreya. So as I said, once you have this hit, right, one of the wells that are hit, that are positive, we run the mass spectrum analysis of that, separate it, and we get the tentative identification of these compounds. And in this case, we got some anthraquinones as a possible antiplasmodial compounds. And through what you call mass-directed fractionation, because we know the mass of the compound, we use our mass-directed HPLC or semi-preparative HPLC, focus on those targeted compounds, eventually get them, confirm the structure to NMR, and finally get the compounds. And once you know the compounds, you then do the bioassays again, and these anthraquinones showed some moderate activity, nothing potent. Typically what you find, so kind of tenfold less active than chloroquine, which was a positive control. But nevertheless, it gave you the active compound, anthraquinones, which we can then do some further modeling against different targets of the Plasmodium parasite. So we do some in silico modeling just to see how the docking scores of these compounds look like. The second example was the second plant, torreya. And this one actually demonstrates, I don't know if any one of you are involved in GNPS. I'm sure GNPS is, for the chemists or the drug discovery, it's one of the biggest databases used around the world. So it's an open source database that you put mass spectral data, you put your biological data, and it'll pop out possible chemical structures of what your compound may be that's giving the activity. So we did that. So this plant, we actually did that. We put in the mass spectral data, we put in the antiplasmodial antimalarial data, and it popped out quite a few compounds and said, these are your possible compounds. We then went about saying, well, let's purify them and confirm that they are. And lo and behold, none of them actually turned out to be right. So they were all novel compounds. Well, not all, but most of them novel compounds, highly complex, what you call liminoids. And eventually got most of these liminoids out, got them tested, and one of them had to be sort of biologically active, not potent as chloroquine again, but actually got to the compound. So the lesson from this is basically GNPS is great, but if you're working with novelty, you will not find it in GNPS. Okay, so that's what I was getting to in terms of chemical diversity, is that we do find that South African plants have a greater chemical diversity, but depending on where you collect it from as well. So at the time when COVID was around, a few years ago when COVID was a major problem, we also said, well, can we look at something within COVID as well? And we selected some 20 plants, and if you do the maths over there, it turned out to be quite a few number of samples that we actually had in the database, put it through a few biological assays, mainly the one, the AC interaction against spike protein. We looked at the disruption of that, and then followed up with that, what the compounds may be to against COVID. Okay, so that's in terms of what we did. Sent up all our samples, looked against the disruption of AC and the spike protein, and 3 plants actually popped up as good activity. One of them was Pedocai Henkley, another one Gunnera papensa. One I haven't showed you is called African ginger, which I'm sure many of you will know about. And that actually gave very good spike protein interaction activity. We put that into a cell-based, infectious cell-based assay against the Wuhan virus, and we actually able to identify that this enzyme assay was replicated in a cell assay. So it's actually working on the live virus. So a good case study from this was actually African ginger is one of the most commonly used medicinal plants in the country. And we put it through the same assays against COVID. So cyphenocolone is a chemical compound that's well known and well published to occur from this plant. And because we followed a different approach and we were able to fractionate and identify the minor compounds, we identified another compound which we called HLS, kind of oxidized form of this, identified this compound, which turned out to be 3 times as potent as cyphenocolone and completely non-toxic. We got to a point where we actually patented this. For COVID, it worked against all the strains, the Wuhan, Delta, and the Beta strains, and was comparable to remdesivir, which is a conventional drug that was used for COVID at that time. So we filed a patent, but as you know, COVID's not really a problem anymore, so we should be publishing this data soon. But again, the lesson from this is because we actually use a high throughput purification and able to detect very small small quantities of compounds and very sensitive, we're able to identify the real active and not what is commonly known in this plot. We did some high throughput screening against prostate cancer. So you may know prostate cancer is one of the major cancers that is still relatively untreated. And there we tested over 300 samples against the prostate cancer cell lines. This was due to the Curie Institute in France. And because you have high numbers, we have to present our data kind of differently against different cell lines, different concentrations. And in this case, we found just only 5 plants or 5 samples that were quite active against the prostate cancer and commonly to all different cancer cell lines. We then went about to identify in those 5 samples, and one of them we were fortunate to identify this compound called sclerothrin from the plant Xanthoxylum capensa. And one of the fraction F6, as we call it, after semi-pure, was almost pure. So using our technique, we got a compound that was almost pure, and it turned out to be sclerothrin, which when we tested in the same cell lines was highly potent against prostate cancer. So it had very good activity. But unfortunately, the compound has been thoroughly researched by others. In fact, it was found as one of the protein kinase C inhibitors against triple negative breast cancer cell lines. Been well published. It's also been into clinical studies. So it basically showed that our method does work. But again, we came to a compound that was already published and unfortunately could not take it any further. So in the process of just publishing the data. We also you can apply this technique to metabolomics as well. So that's kind of a fast growing area. If you look at the literature recently, a lot of it is on metabolomics. So we looked at anti-tuberculosis screening. So we screened a lot of samples, again, over 500 samples that we screened against MTB. And what we found in this case is certain samples were highly active and some samples are not. So when you're talking about samples, we're talking about specific purified samples. It's not the crude extract that we're working anymore. And because they are semi-purified, they're less complex. Those in the green, we actually identified as active. And we have used the metabolomics approach in this case. So we run an LCMS of each of these samples. And through multivariate data analysis, we're able to then predict, well, the software is actually doing it for you. It predicts the structures or gives you the mass spectrum of the compounds that are highly probably to be active. So that's the chemometric analysis and the multivariate data analysis that we are able to get to the compounds without having to purify them. Okay. And that's because of the mass spectral data that we are able to generate. And once we have the compounds, we're now busy with some docking studies against the enzymes. And before we actually go in to actually see if they actually work in a cell system itself. Okay, so that's in terms of TB screening. One of the highlights of our work has been for HIV. So we worked on several plants for HIV, and one of them came out as this plant called Cnidia sericiocephala. And this compound, this extract of this plant to be highly potent against HIV. And using the same techniques to purify, we eventually came up and purified the chemical compounds responsible for anti-HIV activity. And of course, we purified this again, and you can see the compound chemical classes, what you call daphnane diterpenoids are highly complex. So we did come up with at least 5 compounds, one of them being novel, purified them in sufficient quantities. Again, going back to chemical diversity, we seem to find good diversity in South African plants, provided you're not just looking for flavonoids, because that's the other thing that's coming up very commonly. If you just follow the general approach of fractionating using your silica chromatography and the way it's been done, everyone comes up with flavonoids and your terpenoids. So using these different techniques, we seem to find better diversity, especially these complex compounds here, daphne and diterpenoids, which when we tested against HIV, and this is in the cell infectious cell assay against subtype C, we found the activity to be very equivalent to tenofovir, right? So it was active at what you call 30 nanomolar with a selective index of greater than 500, very equivalent to tenofovir, and even more, it actually has the ability to reverse HIV latency. So if you know, HIV also goes into a latent stage, and that is the big problem, is that once the latent stage, the conventional antiretroviral drugs do not get to latent virus until one day the virus then decides to start replicating again, and that's when the person gets AIDS. So this has the ability to reverse latent HIV as well. So fortunately for this, we've actually, don't think it's in my slide here, we've got funding from the Medical Research Council and we've moved this into preclinical studies. So it's going to be going into animal studies soon, and hopefully we can get this into clinical if it's proven to be safe in the next stage as well. And I think that's my last slide, but other than just to thank some of my partners. So I must say most of my partners in biological screening have all been overseas. From Oxford University to the United States to Switzerland, India, China. And the only reason I give it to them is because they have a high throughput screening methods. Unfortunately, we do not seem to have that in South Africa, other than Institute for Malaria Control, which is at University of Pretoria. Others don't seem to do. So if I give them 10 plates, 96 well plates containing the samples, no one's able to do that. So I've done most of this internationally. Yeah, and so yeah, that's basically, that's kind of where I am at the moment. Okay, thank you.
Okay, thanks very much, Professor Maharaj. And that's fascinating to see these case studies where you really get to the final products. Let's open the floor to questions.
Thank you, and I agree, fascinating workflows, but obviously very human orientated, which I've tended to avoid in my preference. So I'm going to, I guess, err my question towards my bias. Could you use this particular platform to extract compounds that, say, could then be applied to farming? An example being the great push towards sustainability, net carbon zero, controlling methane gases through feed treatments. Is there scope to use this in the same manner as you do for humans, but for agriculture?
Okay. Yeah, thank you. Two, I wouldn't call them case studies, but 2 studies that I have at the moment. One is actually looking at the library that we're testing against some pathogens against citrus. So we are doing that. And basically the approach is exactly the same, except in the case for the citrus industry and the pathogens there, we find that we need larger quantities. That's kind of just an outcome that we're finding, but maybe because it's a different organism human compared to a plant. The second one is an interesting one. We're actually doing a study with the Department of Animal Science. Karina's left. Yeah, so they look at enzymes, and some of the enzymes that actually do the conversion actually to methane gas. So we are testing some of the samples to be able to stop that conversion or reduce that conversion to reduce the methane gas emissions from cattle.
Another question there, Jasper.
Vinesh, the original collection of these plants is a long time ago, if I remember rightly. And I think you said there were 11,000 samples, but I think you've told me there were 7,000 species of about 25,000 plant species in South Africa. Was there a selection criteria when they were collected? Or was that, I hesitate to use the word random, but guide us.
So the collection is old, yeah, but fortunately it's in a dried ground stored form. So it's all stored in the ground form, and we've shown that, well, not we've shown, but quite often you find in terms of research, when your plants are stored dried and ground, they're relatively stable, unlike when you actually make an extract. So in fact, we did take all of them and we made 32,000 extracts. And we had, at that time at the CSR, tested all 32, not tested all, but we started to test these 32,000. But because they are plant extracts, they're so complex. And once you get a hit, you just cannot do anything else, but other than to convert it into a 2-3 year PhD study. We dropped that whole activity. All of the 32,000 we had to discard because they are unstable. So we lost all of that, but the plants themselves are quite stable. They do represent some 7,000 plant species because, every plant you'd collect the leaves, stems, and roots separately. And your last question is in terms of how is selected. So I would like to use the word random, okay, because we just sent the collectors, many of them from Sandbees, yeah. They just went around the country and started collecting. So some in the Northern Cape and Eastern Cape, and just collected. The challenge we had, of course, at that time, we didn't have technology, and we never knew that we were all collecting the same species at times. So they would all bring a Lipia species from the Northern Cape, from the Northern KZN, from Limpopo, from the Eastern Cape. And we had to stop it because there were too many replicates that were coming along. So it meant to be random, and we didn't have any really criteria, but some of the times we actually did say the criteria for the collectors was to go to unique biomes. So the biomes was maybe a bit unique. So a lot of plants did come from the desert because been told that the ancient plants seem to have more biological activity.
Yes, the question here. Okay, how do we access the online question? Can you access the question? Can you see it?
Should be in Zoom somewhere. There's Zoom.
We note your question, that is, your hand is up, Tobias. If we could just ask somebody to unmute him and share his.
Okay, they can just ask you.
On Zoom. There's no Wi-Fi. So not on Zoom.
Yeah.
Able to.
Yeah, thank you. Thank you, Prof. Professor Tobias Ezejofor from the Federal University of Technology, Owerri, Nigeria. I think I've listened with keen interest to your great presentation. I'm thrilled by the great extent you have taken these plants in their search for their activities in various disease conditions. Here in Nigeria, we are also blessed with a great diversity of plants. And we, as part of our desires, have been trying to find out indications for some of those plants, what they can be used for. But unfortunately, we are not blessed with the kind of technology I've seen applied in your studies here. So the question now is, can we be granted the grace to work with you and able to, for some of my PhD students working in those kind of studies using plant species, to see how much we can tap into your knowledge and expertise, and especially the technologies available to you to advance our studies. Thank you very much. And congratulations for the wonderful work you've done so far.
Okay, thank you. So yeah, I've got 4 students from Nigeria who managed to get funding for themselves in terms of bursaries, and they're all busy with their PhDs and graduated one, in fact. So they all actually come over with their plants and the samples and looking at a specific disease. So they spend 2-3 years of the studies in our labs, and they get access to all the technology. The only challenge is, of course, they need to get a bursary for themselves for the 2 to 3 years. In fact, the student who's working on for the citrus pathogens is from Nigeria, and he's brought his plants, but he's also using our plants as well that we are testing against. So yes, it's possible.
Okay, in that case, I would like to have a contact. I don't know how to do it, whether by email, or I would like to have your contact in order to take discussion beyond here.
So is it not in the book?
Yeah.
Yeah, I thought it would be in the book of abstracts. Is it not there?
But you can Google it.
Ah, you can. Okay.
Pardon?
Thank you.
I'll put these contacts in the chat box so that you'll have access to it.
It's okay.
Okay, thank you. Right.
Thank you.
A question there from Olivia. Oh, there was actually one over there. Let's go there.
Okay. My question is, how are you partnering with those people who discovered medicinal plants? Not necessarily discovered, but those people, are they called traditional healers? Because usually they are the ones who has more knowledge on the usage of medicinal plants, irregardless of their toxicity level, because some of them, their patients end up in the hospital due to toxication in their livers and all those kind of things. So as someone who has both history of science and health, I'm more interested in the medicinal plants used to treat sexual transmitted diseases, because I do know that there are some things that cannot be treated with the Western medicine, but they can be treated with an African medicine. So how are you using your library to partner with those people? Because even with my study, I had to go back home and consult with an elderly woman who has been doing this for years on how can I use her plantations for my study. Thank you.
Yeah, thank you. So my group belongs to a consortium that is governed and controlled by the Department of Science and Innovation. And this consortium basically looks at protection of indigenous knowledge and utilization of indigenous knowledge. So most of the knowledge holders would actually approach the Department of Science and Innovation, and through them, they then refer the knowledge holders to us to actually do the research. So that's actually how we work. While I must say at the same time, there's a vast amount of literature that's published on a lot of the use of medicinal plants. As an example, in South Africa, you pick up a few books, you'll find a lot of information already published. So we also use lots of published information. But at the same time, South Africa is also governed by the Biodiversity Act. So we have an act in South Africa that governs any use and commercialization of indigenous knowledge, that you have to collaborate and work with traditional healers or knowledge holders. So that's the way we actually do it. Thank you.
Okay, let's have one last question. Olivia?
Thanks, Prof, for the presentation. Very good presentation indeed. I'm curious in terms of how we are planning ahead in terms of either breeding strategies for these indigenous plants that we know that are useful and we're gonna need it for the medicinal purposes. What are the plans
in terms of us conserving them, and how are we assisting the community to be able to breed better, because at some point these species are going to extinct and we're going to end up with none. And how do we best start to, now that you lose some of the collection, how do we go back and start to gain back these collections so that we're able to now explore them and do the work that it's not yet done, or building up this genome reference so that it allows you with the research that you're doing to unfold those interaction or the molecular understanding around it.
Yeah, in terms of the genome interaction is something I would like to do, but I have no expertise in it. And I've always spoken to some of the experts to maybe want to do that. And many of them say it's a bit too complex with plants. I don't know, real or not. So
it can be done.
It can be done. So, but looking specifically at cultivation, I work closely, very closely, in fact, through the consortium again with the ARC, mainly in Ruderplaat, and they actually do all the cultivation. In fact, one of the plants that we're talking about in terms of cultivation, the two that are actually moving into preclinical studies, one is being cultivated in UKZN, and the other one being cultivated in Mpumalanga. So, but that's done to the ARC. So we give them, because they're experts in this, in the cultivation, we hand that over to them to be able to do the cultivation, especially for commercial purposes.
Okay, thanks very much. I think we should have put in a lot of time, extra time for discussion here. But I think Professor Maharaj will be around at the tea time after this. So let's keep if you can keep your questions for then, and so we can keep on with the program. Thank you very much. I think let's have a round of applause. It was fascinating and lots of questions. Okay, so if our IT people can help us to connect the presenters from Zoom, please. The next presenter is P. Kelechi, a young scientist speaker. He's going to be telling us about marker-assisted breeding in rice using SSR markers. So if we could just call up presenter online.
Hello. Yes, sir. My name is Kelechi. A graduate of Corporational Scap Management University.
Hello, Mr. Kelechi. It doesn't sound like the sound is working so well.
My name is Parker.
You can check there at the IT. See to some feedback there.
Can you hear me?
We can't hear you so clearly. Have you shared your screen with your presentation? It's just apologies for this. Let's see what they can sort out there at the back. We're going to try and share the presentation here, and then he can just speak on his phone maybe without the presentation. Hello, Mr. Kelechi. Can you hear me here? We'd just like to request that you only put on audio from your side. Please turn off your video, and then we should be able to hear you better. Do you want to try that? We've got your presentation on here, and you probably won't be able to see which slide it is, so you'll have to just tell us to move from one slide to the other. Is that in order?
Yes.
Okay, do you want to go ahead and your first slide is up here, and you must just maybe just speak very clearly and slowly, and then just tell me when to move on to the next slide.
Awesome.
Okay, so your first slide is up.
African Genome Project Open Institute South African Regional Workshop 2023 presentation by Paul Kirch, a graduate of Production and Landscape Management at Ebonyi State University. On the genetic diversity and stability analysis based on morphological trends among us genotypes. So that's
Hello, Mr. Kilichi. I think the sound is just not good enough. I don't think people can hear what you're saying. So no, unfortunately, I think it's just not clear enough.
It's not clear now. Can you hear me now?
Yeah, your location is not very good. There's a lot of background noise. You can't move to another place. And are you able to move to a quieter place?
Yes, that.
I think that we're going to have to call off your presentation. Do you want to try?
Yes, it's better now.
Okay, that's much better. Thank you. Okay, so we're on the first slide. Shall I go on to the next slide?
Okay, the next slide.
Okay, next slide is on your abstract.
Yes, sir.
And if you could try to use the minimum words on each slide, just because it's not that clear. Please go ahead with slide 2.
Okay. Abstract. Background of the study. Capacity and stability analysis based on morphological stress and genotypes were developed through marker-assisted backcrossing. Was evaluated the collection of references as Ebonyi State using this study.
Mr. Kilichi, unfortunately, it really is just not clear. I think the technology is just not good enough to use a phone, unfortunately. So I think we're going to have to call off your presentation and go on to the next one. I'm sorry about that, but we just can't hear what you're saying. Okay, apologies from this side. So if the IT people can call off the next presenter. So Mr. Kilichi, we'll talk to the organizers and see if there's another opportunity for you to present your work. I'm sure there'll be other opportunities at other forums, and we're very interested to hear about your work. So let's set it up for another workshop, if that's okay with you. Okay, so I think that's a good, this is a good learning experience for the Africa BioGenome Project. You know, we have to understand about the technology constraints that people have, you know. So so that's something which needs some attention. But I think I know from my experience it's I can't use my phone to tap into a Zoom call here in Pretoria. You know, if there's an institute presentations, the phone is just not good enough, you know. So right. Yeah, I think it's just the amount of bandwidth. So with the next presenter, do you think they can possibly get online? Okay, so the G by Sese, is that person here? So they're not an online presentation. Does anyone know by Sese? Talking about cooperatives on honey household incomes and forest conservation. Maybe that's also online, or is it, you don't have a record of whether it's online? Is it supposed to be online? Oh, here, there is a presentation here. Is Southeastern Kenya University. I've got it on here. Hello, Mr. Sese, are you online? Are you listening? There's a presentation here for sure. So it sounds like you most likely would be online because it's from Southeastern Kenya University. You don't perhaps like have a cell phone number for him or anything like that? Yes. Okay, fantastic. Is the next presenter here? Okay, so Kay Sabetse, exploring plant insect interactions at Pretoria National Botanical Garden using DNA barcoding, remote sensing, and statistical modeling. And that's wonderful that you're here. Let's see if we can get your presentation up here. There we go. Okay, so if the IT people could please just put this up on the screen. I've got it up here. So it's Cabo, is that right? Yes. Okay, welcome. All right, so you can sit here and that's the microphone there. If it's red, then they can hear what you're saying. Can they connect this to Zoom? Can you connect this to the screen, please? You're welcome to. So you're close to that. Okay. If you speak into the speaker, it doesn't seem to be very good. Oh, okay, so okay, good. Okay, so we need to just put this on slideshow. Okay, great. Okay, should be. Okay, it's on slideshow now. Okay. All right. Good. It's getting fit. Wonderful. Okay. Over to you, Kaba.
Thank you. Good afternoon, ladies and gentlemen. My name is Gabo from the University of Johannesburg, and then I'm honored to be presenting to you my research. So biodiversity on Earth is sustained by different species interacting together, forming complex web of interactions that influence the biodiversity as well as the ecosystem health and stability. So among these interactions is the relationship between plants and insects. So plants and insects, they form the important component of terrestrial ecosystem, with plants serving as the primary producers and the insects occupying the different trophic level. So their interactions, like they range from mutualistic interactions to commensalism, parasitism, and predator-prey relationships. These relationships, they support important ecological processes such as herbivory predation, nutrient cycling, as well as pollination. So in Southern Africa, where agriculture plays the key role in the rural livelihoods and food security, pollination, it serves as an important ecological process that boosts agricultural productivity by contributing to the production of 70% of the crop species. Many of the high value food, such as the almonds, the berries, the apples, and coffee are few of the example of food that are, or the crop plants that are dependent on insect-mediated pollination for reproduction. The process of pollination, it also relies on the synchrony between the flowering times as well as the emergence of their pollinators. However, with the recent major decline in the insect species, as well as the increase in global climate change and anthropogenic activities, the synchrony between the flowering times and the insect pollinators is being altered, with some of the plants flowering more earlier or later than the emergence of their insect pollinators, leading to reduced pollination success as well as crop yield and quality. So given these challenges, it is therefore important to study and conserve these interactions to sustain the ecological processes they provide. So for my research, the aim is to employ the modern surveying techniques, which is the remote sensing as well as statistical method to explore these interactions at Pretoria National Botanical Garden. And the objectives includes to determine the temporal changes in plant phenology using remote sensing, to determine the correlation between the plant phenology and the abundance of insect pollinators, as well as to compare the insect abundance in the cultivated and the natural areas within Pretoria National Botanical Garden. So it is also worth mentioning that what I'll be presenting here just forms part of my first objective for my research, as my research is still in its early stages. So this study was conducted at Pretoria National Botanical Garden, whereby 2 sites were chosen for comparison. We have the cultivated and the natural site. These sites were also chosen for their difference in ecological characteristics as well as their management practices. So for the data collection of the plant phenology, we used remote sensing to obtain the monthly plant phenology around the vicinity of the Malaise traps by calculating the normalized difference vegetation index around the 10 meter by 10 meter quadrant around the Malaise traps, and then use the machine learning for the extraction of valuable information about the timing and the duration of the flowering events at a larger scale. Then for the data collection on insect pollinators, the data was obtained using the Malaise traps placed at the cultivated and the natural sites throughout the year in order to capture the seasonal variation in the seasonal variation also in the diversity of these insect species. So the design of the trap is such that the insects flying in the vicinity of the trap are funneled into the collecting container, which includes like a preserving alcohol to preserve the insect sample for further analysis. So then the collected insect species were then subjected to DNA barcoding pipeline, which includes the DNA extraction, PCR amplification, DNA sequencing, and the bioinformatics pipeline. And then the insect functional groups were then classified using the text mining and the natural language processing, whereby over 30,000 articles were used to assign these functional groups. And then for data analysis, the latest version of RStudio with its relevant packages were used for data analysis and model construction. For this presentation, I won't be presenting much more on the statistical models as I'm still busy with them. So for this, it represents the normalized difference vegetation index, the NDVI map around Pretoria National Botanical Garden. So the NDVI actually, it assesses the vegetation health, as well as it can also be used to measure the plant phenology. So the darker greener areas, they represent like the healthier and more dense vegetation, whereas the lighter green to yellowish areas, they represent the non-vegetated areas. So then using that data, then I calculated the NDVIs to plot this graph to compare the plant phenology between the different sites throughout the year. So for the two sites, the cultivated and the natural, they sort of like have the same trend throughout the year, but there's that deep in the month of March. I hope you can see that. And then it then increases in the month of April, then decreases during the winter months, and then it increases during the spring and the summer months. So this dip in March, it can be explained by this this decrease in the precipitation level that were recorded throughout that month. So this also shows that the environmental variables such as the precipitation and temperature do play a role in also influencing the plant phenology as well as the insect activity. Then for this one, it represents the different insect functional groups that were obtained from the insect samples, so as to capture or to show the diversity within this class. So with insect herbivores, which are those that feed on plants being the most abundant, followed by the processors, the parasitoids, and the pollinators. So I'll be mostly just focusing on the pollinators, as I'm still busy with also like generating results for other insect functional groups. So for the pollinators, out of the 51,234 insect sample at the Pretoria National Botanical Garden, there are 10,898 pollinators within the whole garden that were sampled. And then this then represents the major orders that were found within the garden to show like the diversity of these insects within the garden, with Hymenoptera being the most dominant, which includes your common pollinators such as your honeybees as well as the wasps. So then to graph out the relationship between the plant phenology and the insect pollinator abundance, this graph shows the relationship between the two, whereby a seasonal correlation can be seen here. They sort of like follow the same trend throughout the year, whereby it can also be seen that the plant phenology measured as the NDVI influences the insect activity throughout the year. And then for the natural side, from the month of January to June, there is that, for the blue line, there is that rapid decline in the insect pollinators, even though the plant phenology relatively remains the same. So this shows that other than the plant phenology, there are other factors that also influence this insect activities, such as maybe it could be like their own life cycles. And then for the cultivated side, it's also for this one, there is just that major decline in the insect pollinators, and then just a sort of like a correlation in the later months of the year. So with this one, it represents the density plot that was generated using over 390,000 images to show the mean flowering time throughout the year, with that green line representing the main flowering times, and then the blue line representing the pollinators. So this is sort of like show like the interaction or the correlation between the two, whereby mostly during the winter months they are relatively low and then start to increase during October, and then with the end of January to early February being the time whereby most of the insect pollinators are most active. So in conclusion, the plant phenology at Pretoria exhibits distinct temporal patterns between the different sites, and that the insect activity is influenced by seasonal variation in plant phenology and environmental factors, and that the natural area exhibited a high insect diversity and abundance. This is due to like the less disturbances due to like the management practices. So my future work is to work on like the other interactions between the other functional groups with the plant phenology, as well as to use the predictive models to predict like the effects of the environmental variables on the plant-insect interaction, as well as to construct also the statistical models to look deeper into these different ecological interactions and also the common patterns that drive them. So for the acknowledgement, I would like to thank my supervisors as well as the center I'm affiliated with, the African Center for DNA Barcoding. And then also, I forgot to include also the Center for Biodiversity Genomics in Canada for helping with the sequencing of the large data, which I hope that next time we will be doing our own sequencing within our country. I thank you.
Okay, thank you very much, Kabo. That was nice to see how you're trying to use data to deal with identifying 25,000 insects in one sample. Questions from the floor? There in the front, please.
Yeah, yeah, nice work. So I noticed that the insects you're capturing are flying insects. And so what I'm wondering, do you get any crawling insects that go into your traps as well? And would you expect some differences in the relationships you're seeing with flying versus crawling insects?
Okay, yes, most of this data, it's mostly from the flying insects, but what we have also tried to do is also like to include the crawling insects. There are crawling insects, but there's just a few abundance of them. So there is just much abundance with the flying insects more than the crawling insects. So I think the reason mostly of we included like the flying insects, it's because of like how they are able to sort of like let's say they can be sort of like give us an idea of how like the climate change also like affect their, let's say their movement, like like maybe like within the garden. So how like they sort of like move. So that's why mostly it's just the flying insects instead of like the crawling insects.
Agree, great work. And perhaps as an extension of that, I wonder if you'd considered leveraging the power of sequencing and indeed that of eDNA to sample the flowers that have been pollinated and residual DNA on the flowers to then match with the insect species that you had trapped.
Okay, so what I did actually from last year, we did this field observations whereby we did sort of like capture the like we went to the garden to capture the data on the flowering plants. So, but then we haven't applied any of the lab work like the eDNA or the meta DNA barcoding on that. So, but that's what actually I'm planning to do for my PhD, to delve deeper into this, because what we have mostly focused on is the insects more than like the plants. So yeah, I think in order to understand deeper like this interaction, we will need also to focus on like the DNA barcoding or the eDNA of these also plant species.
Can I just ask a question? So I'm just curious, what were you using to identify the insects? Were you basically putting all the insects you got in one and just crushing it up and making one DNA extraction and sending it for like Illumina sequencing with the PCRing up with the barcode COX-1 gene and then sending it off? Or how were you, or were you using images of the insects?
It was
I'm not quite sure how you were actually able to actually identify the insect functional groups.
Oh, it actually, we just also, it was through the DNA extraction of these insects. So that's one of the reasons like they were sent to like the Center of Biodiversity in Canada, because we don't have like the resources to be extracting like the DNA from each and every single one of them. So what we did, we just took the data from the BOLD, the barcode of life databases. So that's what we did. And then, as I've mentioned, using then the text mining and the natural language processing, and then we then assigned the insect functional groups to these using the bioinformatics tools.
Okay, so did the collaborators actually sequence each individual, barcode each individual insect separately?
Yes, they did.
Okay, it's interesting. But I'm wondering why you couldn't have just PCR'd up from the bulk and then just put that through Illumina sequencer. Maybe that's something to think about for your PhD. Okay, any other questions from the floor? Yes, thank you.
Thank you for an interesting presentation. Maybe my question will be a bit different because when you talk plant-insect interactions, I'm already thinking of the beneficial aspects to say when the insects go into the plant, maybe they introduce some microbes. Have you got any interest in looking at the health of the plant in terms of what happens when the insects go into the plant? I don't know, I'm just thinking of that. Thank you.
Okay, that's also one of the things I'm trying to also look into. So as you've seen, there are other sort of like insect functional groups. So sort of like the processors and the ones like the insect herbivores. So I will also be looking into, because there are plants that we sort of like sampled. So I'll also be analyzing like the sort of their health as you and then like the damage that the insect herbivores they have caused on this plant species. So it will just be for that part, I'll just only be focused on the insect herbivores and the sort of like the damage that they have done on these plant species. If that couldn't be done, I will also be looking like through the herbarium specimens that we actually did last year for these plant species to actually study these, like how the insect herbivore cause like these damages to the plant insects. Yes.
Okay, thanks very much. Any other last questions for Kaba? Okay, let's give him a round of applause and good luck with your studies.
Thank you.
Okay, so if they could maybe, is there any chance that that one speaker is available? Talking about the honey household incomes, or are they not available? Should we skip that talk and go to tea now, and then we'll end off with the last session? Is that okay, Olivia?
It's still available.
In the next
second
session.
last.
Okay, and then after Prof. Cuthbert Banga was supposed to be chair the next one. Is he here?
Dr. Masebe is chairing.
Okay, you were going to chair it. So, and the one speaker we were thinking about was Mr. Noma. You are here. He's here. Okay. Okay, so we've got one speaker, then we potentially got one online speaker if the technology is okay. So should we take the break for the tea and then?
Yeah, just 10 minutes or so.
Okay, the time now is 4:15. If we can come back at 4:25 for the last session.
Yeah.
Okay, let's give a round of applause to everyone.
Can I also take photos for everyone now? I forgot in the morning.
The new P4 flow cell. Experience more reads, greater accuracy, and the most cost-efficient runs on NextSeq 2000. Expand the utility of your benchtop system with 4 new run configurations. Maximize batching at scale. Applications like exome, single cell, and spatial sequencing on the market-leading platform. Do more with P4 on the NextSeq 2000.
Quang tên khỏi quang thuộc cả nó phát quang tên họ thuộc cả này quang tên họ thuộc cả này phát nó quá thì bệnh quá thì hùe cũng thay chế mà phần nát mà lúc tê cài lắng ăn chế ai chào hùe lắng bọn này chế ăn chế léo bọn này chế.
It's projected that the world's population will grow another 2 billion people in the next 20 or 30 years. These fish look in really good condition. The key thing here for farmers is to be able to produce enough fish to feed the population in his area, but also globally. My name's John Benzie, and I'm the Acting Director of Aquatic Foods Biosciences for World Fish, based here in Penang, Malaysia. Fish are highly nutritious and in many parts of the world are the key components to people's nutritional security. It's important to produce more of these foods. One, because they're nutritious and they've been fed today, but secondly, because there's a huge demand for them that is not currently met. In this hatchery, we maintain the genetically improved farm tilapia strain known as GIFT. This fish now grows twice as fast as unimproved tilapia.
Let's try. Yes. Yep. Okay, so let's go.
When the fish are large enough,
slowly,
we take a small piece of the fin. Doesn't harm the fish at all. And we take that sample to provide to our research partners in the UK, Errolham Institute.
Hey guys, Where we come in as the Earlham Institute,
You've got lots of nice
what we do is we look to sequence the genomes of these fish. It's quite a small fish, and then annotate the functional and non-coding aspects of the genome to then better inform how they could then improve these fish further.
We could utilize the ability of the Nile tilapia to grow particularly well and fast.
I'm Karim Garbi. I'm head of genomics pipelines at the Earlham Institute in Norwich, England. The issue that we're trying to tackle with this collaboration with WorldFish is to try and improve aquaculture in Africa and Asia of tilapia fish. To do this, we use state-of-the-art technologies to look at the genome as a whole, and in some cases, very specific areas of the genome that might be of interest and linked to these traits that the farmers are interested in. So the benefits of the NovoSeq for this type of projects are twofold. The first one is reducing the cost. The second benefit to us is the speed.
What's really important there is that allows us to apply multi-omic approaches, which gives us better power to translate some of our findings to deal with my mission of improving food sustainability, but then also Earlham Institute's mission to also protect life on Earth.
This is my passion, my scientific passion really. So being able to connect the technology that we've set up in this lab to the life of farmers and the livelihood of farmers, and helping them thousands of miles away with the work we're doing here in the lab is fantastic.
It's going to be important to produce not only organisms that grow faster and produce food more quickly, but also do so with less environmental impact. Feels like a worthwhile thing to do, and it's satisfying at the end of the day to think that you've made at least some small contribution to that process.
For over a decade, Illumina's solutions have enabled researchers and public health scientists to make groundbreaking discoveries and protect our communities from infectious diseases. From enabling the adoption of whole genome sequencing to aid investigations of foodborne outbreaks, to more recently helping public health officials around the world track the transmission of SARS-CoV-2, Illumina has been dedicated to those studying the smallest and deadliest threats to human health. Today, we are proud to introduce two new library prep solutions built on the simple and affordable COVID-seq workflow that will help build global genomic surveillance capabilities and power impactful microbiology research. Illumina Microbial Amplicon Prep, or IMAP, is a unique library prep solution allowing researchers to use lab-designed or commercially available primers for targeted sequencing of viruses and select markers from larger microbial species. Ideal for One Health applications, the kit provides all the reagents needed to go from extracted nucleic acid to sequencing-ready libraries. Illumina Microbial Amplicon Prep Influenza A/B, or IMAP Flu, is optimized to provide extensive sequence coverage across both influenza A and B viruses, compatible with RNA inputs extracted from cultures or nasal swabs and washes. IMAP Flu delivers exceptional data quality, flexibility and scalability for public health surveillance and high-impact influenza virus research. Both solutions seamlessly integrate with the Dragon targeted microbial app. This powerful app serves to simplify data analysis, making it easy to perform consensus genome generation, variant identification, and variant interpretation. IMAP and IMAP Flu, two exciting new additions to Illumina's portfolio of solutions for public health surveillance and microbiology research. Because in the genome era, Infectious diseases don't have to be a disaster.
Human beings have been using astronomical tools to adventure into the infinite universe, to explore the infinitesimal by making use of life science tools and technology. Founded in 2016, headquartered in Shenzhen, China, MGI is committed to building core tools and technology to lead life science through intelligent innovation. MGI has established 7 international R&D and production centers and is mainly involved in 3 key business segments, including genetic sequencers, laboratory automation, and new business. Continuing heavy investment in R&D, MGI has developed a full range of patented technologies, including DNB-seq sequencing technology, patent array technology, and sequencer optoelectromechanical system technology. Gene sequencing has a very high barrier to entry as it involves multiple fields, including electronics, fluid dynamics, molecular biology, chemistry, nanoscience, and other disciplines. MGI is one of few companies in the world that have the ability to develop and mass-produce low, medium, and high-throughput clinical gene sequencers from GB to TB. Besides gene sequencing, MGI has also developed a series of core technologies, including library preparation and automated sample processing. Expanding into new business, MGI has developed multiple innovative life science tools and technology, including portable single-cell systems, remote robotic ultrasound systems, BIT products, and low-temperature storage products, with a strong focus on the upstream of the industry. MGI operates in more than 100 countries and regions, serving more than 2,800 customers to date, empowering the downstream customers in reproductive health, precision cancer diagnosis and treatment, infectious diseases, food security, agriculture, forestry, fishing, and consumer genomics. Since the establishment, MGI is driven to disrupt the status quo and accelerate the industry innovation. MGI aims to build a suite of life science core tools to address the fundamental questions, push the limits of science and technology, and lead the innovation in the era of life.
Afternoon, colleagues. Can you hear me? No? Good afternoon, everyone. Can you hear me? It's been a long day. Yeah, I think it's been a very interesting day. Hearing our keynote speakers, our researchers, giving us more insight into the type of work they're doing in the line of genomics, in the line of looking for potential compounds that can be used for treatment of infectious diseases. I think as we wrap up the day, it's interesting, it will be interesting to know what kind of impact will our research have? Are we just doing research for the sake of doing research, or are we doing research that is going to have impact at the end of the day? I think to sum up that, we have Mr. Noma Ayanda, who will talk to us about capitalizing in IP protection and commercialization of indigenous species and the potential impact on the bioeconomy. The floor is yours. Thank you.
I'm not sure if I should say good evening or good afternoon, but I think it suffices to say good day. So I'm just going to connect. Just going to connect my presentation. As they are connecting, let me just introduce myself. Said I'm Ayanda Noma. I'm the Director for Innovation, Technology Transfer and Commercialization at UNISA. And as the presentation is delaying, I may be forced to say certain things that I don't really want to, but let me just keep quiet until the presentation is. But I'm really glad to be here. Oh, the presentation is there. Good, thank you so much. So I know that there was a topic that was mentioned. I'm not sure if I'll speak to all of those, aspects. Is it there? I'll speak to all of those aspects, but I will try. I will try my best. And if I don't, I fall short, you'll forgive me, right? So, I'm used to standing up, so there's an edge for me to stand up, but it's fine. We'll see how this works. So there's no outline. I normally tell people that come with me, let's explore, whatever the conversation takes us. Please feel free to take the conversations in different directions that would be of interest to you. But I'll try very, very hard, just to talk about, a little bit about tech transfer, but also to talk about, some of the stuff that the program director referred to. So I always, talk about, these issues that affect us in our own context, but also I suspect that, it affects the rest of the continent. But of course, in our space, they're probably more so. And you'll ask, perhaps you may be asking yourselves, why are we talking about these issues? But go with me. We'll get to that answer. But you can see, one of the issues that we have, one of the challenges that we have, and I'm sure people talk about the triple challenges that you're aware of, poverty, unemployment, inequality. Equality is one of those who've got the highest Gini coefficient in the world. Highest inequality in the world. And of course, one of the perhaps promises that were made quite earlier, on around the issues of transformation was that would then be able to grow this pie, that is our economy, so that it becomes quite inclusive, but it doesn't happen. And of course, it's a bit of a challenge. You also have, you can see the income per capita has sort of stagnated over a number of years. And of course, there's also a very, very real regional divide. If you are in the Eastern Cape, you're probably in the poorest province, right? I come from the Eastern Cape. I raise my hand. I come from the Eastern Cape. Therefore, the Eastern Cape, if you look at it, right, is the poorest province, in South Africa. And of course, it's cause for concern. I'll tell you why. And of course, if you look on the right-hand side, in terms of unemployment, right? The demographic that is most affected by unemployment is the young people. And I mean, if you look at this is not sustainable. By any way you look at it, it's not sustainable at all. More so when you look at, if you were to consider the prospects, that's how our economy looks, in terms of, the growth prospects. But you can also see, that there's growth in some areas, but in some areas there's a decline, right? Some may say these are structural challenges, but also this presents opportunities for us. There's also this conversation, around sunset and sunrise, in sort of sectors. Which of these sectors are in decline? There are others that should be, that should be on the rise, right? These are new sectors, and where are these sectors, going to come from? Who's going to lead the charge in those sectors? And of course, the expectations. And really the expectations generally across society, that we should all be contributing, but more so, in the higher education space, particularly when it comes to R&D. This is a very old picture. It came from the Financial Times in 2005. And I always use it, I like it, right? Because it reflects, right, the changing expectations on the academy. It says, the ivory tower must now become the watchtower. If we talk about the knowledge-driven society, knowledge-driven economy, who are the people that are at the forefront of this? And of course, we may, there's another slide that I'll show you, we may talk about these things in theoretical terms, in terms of, well, we should have this knowledge, sort of disinterested knowledge seekers that may pursue knowledge in all different directions, that they desire. But of course, there's also that aspect that faces us. I mean, if you look at the expenditure, global expenditure on R&D, look at expenditure on R&D globally. This is the report. This is the latest, science, technology, and innovation indicators report. If you look at Africa as a whole spends about 1% of the global expenditure on R&D, 1%. Of course, Brazil, South Pacific. I'm pointing out, the reason why I'm pointing out this, because also we're part of the BRICS, right? But of course, if you look at our government expenditure on R&D as a percentage of GDP, we have come down, from 0.82, now we're sitting at 0.6. And of course, other countries have continued to surge forward. If you have the whole continent, contributing about 2%, 1%, sorry. And then of course, if you look at India, 2.6%, that's where it's at. I mean, if you look at some of the regions which we are sort of connected and linked to, quite high, China, leading the charge. And of course, as part of the BRICS, it tells us something that in whatever we do, when it comes to, the things that we've got to do, I don't think that we've got the luxury. We have to make choices, right? We really have to make hard choices. And some of these choices, well, not so hard when you think about it, in terms of partnering with others, so that you are able to meet this sort of socio-economic outcomes. And in a nutshell, the reason why I raise those issues, just to demonstrate the point that there's an expectation, and the expectations have been shifting on the academy. The academy now is expected to really contribute to not just to the knowledge creation, not just to students, but also to other socio-economic sort of outcomes, contribute to the growth in the economy. Are there some objections to this? Yes, there are. There are conversations. Of course, these become academic, particularly if you look at what we have to spend on R&D. As conversations become academic, because at the end of the day, the expectation, the public expects that there should be solutions that the clever ones were the ones who are producing knowledge, who are sitting in higher education institutions. They should be producing knowledge that actually translate, into socio-economic outcomes. Hence, you have, the establishment of what are called technology transfer offices, similar to, that when you talk about the technology transfer office, you're talking about my office, the Directorate of Innovation, Technology Transfer and Commercialization. What we really seek to do, well, we were established in terms of a legislation. This legislation is called Intellectual Property Rights from Publicly Funded Research and Development Act. Very much similar to some of the legislations, that are out there. The most sort of famous of these is the Bayh-Dole Act. If you come from the US, you'd know the Bayh-Dole Act. And of course, its sister act called the Stevenson-Wilder Act. That says, well, these publicly funded, institutions shall retain the IP, ownership of the IP, but also shall make sure that the IP is really developed and commercialized. And of course, similar act, IPR Act in South Africa, which came into force on the 2nd of August, says that any IP that is created, that is publicly funded, created within public research organization, those institutions shall actually hold, retain ownership of such IP and make sure that it's developed and commercialized, for the benefit of the people of the Republic. And of course, that led to the creation of tech transfer offices, in almost all, public research organization, including where we are. Besides UNISA, the ARC has got one, and Cynthia is sitting right at the back, if you did not know, of the room. She's responsible for the tech transfer office here at the ARC. So in a sense, as the office, we are responsible for making sure that they are the correct policies, that support innovation, that supports tech transfer, that supports commercialization. And that those policies include the policies on intellectual property rights, the policy on commercialization, also responsible for contract research, consultancies, and research-related services in other parts of the world that will be referred to as sponsored research. We're also responsible for establishing incentives. We do have financial incentives, and of course, we need to now move and make sure that we begin to embed these non-financial incentives in the system, just to encourage sort of this much. We as an office, we're responsible for awareness and training. We've got what we call Innovation Sense. Innovation Sense captures all our training interventions and all our awareness interventions. We run masterclasses on IP, innovation, and commercialization. By the way, they are available. I mean, we have them. They are free for our staff and students. But at the same time, if we've partnered with other institutions outside that say, well, can our people join? I said, well, with pleasure. They are virtual, right? We do have, we train as well. We do have what we call UpTech, which is really a 7-week business model canvas training intervention. We're responsible for establishing partnerships, right? And these partnerships really focus around innovation, with various funding agencies, with other institutions outside of the institution. Of course, I'm touching as well on the internationalization. We also have what we call innovation support, where we've got funding mechanisms to support innovative ideas, not just by staff members, but also by students. Now we've introduced a new program for the alumni, quite interesting. And of course, for students, we tend to have quite a few of these programs that we run, which is quite exciting. We also have, of course, we're responsible for commercialization. We also have a unit which we call UniVentures. UniVentures is responsible, for supporting, those staff and students who say, well, we want to create companies. We are interested in starting up, in creating startups and spin out companies. We provide that kind of assistance, for them. And we take you through from ideation, as you see, really from ideation up, all the way to accelerate. We've got different programs, accelerate, build, and so forth. Ideate, build, and of course, accelerate. Right. We're also responsible for innovation. That's why I wore this T-shirt. I hope you can see it. I hope you can see this T-shirt. It says UNISA Innovation Festival, right? We have this every year, on the last week of November, where we really showcase the innovations from the university. We showcase what the university is doing with regards to the technologies that are available, the kind of capacity and competencies that the university has. But also it's an opportunity for UNICEF to engage and have conversations about all of these matters. What are the commercialization and so forth. But also, it gives a window to people from outside of the university to see all these facilities that the university has. And of course, it's quite exciting for us. All of this is really towards making sure that we drive, ensure that you as the IP creators, you as the knowledge creators, right, are able to have an impact much more beyond just the publications or even the students that we produce. So in a sense, I mean, coming from our university, in a sense, very much aligned to the vision of the university, which is the African university shaping futures in service of humanity. Now, in other ways, different ways, relating to innovation and tech transfer. So we are structured in this way. We've got all those units, Unitech, Unilink. I won't bore you with this, Univentures, Universe, marketing, but this is really how we structured. And at the top, there's a private company called Unisa Innovations. And of course, it's a company through which we do transactions. And of course, we're still in the process. It was established just after 2021. And of course, there's a bit of conversations around this, but this is a company that we use for all these, that we wish to use for all these, sort of transactions. At the moment, everything is done internally, and of course, it's got its own challenges. Universities in the private sector have got different logics completely, right? So this particular slide just shows you the rundown of all the things that we are involved in. Well, as a sample, some of these are not here. I won't, spend too much time, but maybe start here, maybe spend a bit more time here. And talk about, this is really the process, right? And of course, the graphic makes it look like it's a simple process, but of course, it's quite a bit of a complex process. We're responsible for, one, making sure that the intellectual property that we create is identified, is protected, but also it's developed. That is the funding aspect. There's some development around it. We assist you to get funding internally and externally. I've mentioned some of the programs that we have internally and the programs that we have that are there available externally to make sure that that IP is developed. And of course, we market the IP extensively. We've got various ways of marketing. Besides producing technology profiles that we then engage, external or potential users, on the basis of that, or we also produce, we also use extensively other sources such as the Innovation Bridge portal, where we place all these technologies. We also produce booklets. We also attend exhibitions, right, to really market the technologies. At the end, because what we want to do, what we want to achieve is the impact at the end, either through startups, or even licensing the technologies to existing companies. Now, so this is the process that we manage. I'm not going to talk about all the other aspect of that was listed in the previous slides. Just in a nutshell, right? Now coming to the issue that you are discussing, relating to all this genetic material. By the way, it happens to be one of the areas that I'm really interested in, and my interest was really sparked by, well, it was by chance because we happened to engage with some colleagues from Sweden. And then we spent quite some time in Sweden, and one of the things that we were discussing was there was an offering there called genetic resources and traditional knowledge, and that really sparked my interest in this particular area. And of course you about all this legislation that and of course conventions. You are the experts in these areas, particularly on the CBD, right? Really to the object is to conserve, biological diversity and ensure that it's sustainably used, and to ensure that there's fair and equitable sharing of benefits. One interesting thing, while we were there, I had a friend from, I have a friend from Zambia, as we're in Uppsala. You know, there's a garden, those who know Uppsala, there's a garden there. As we were walking in that, Linus's garden, as we were walking in that garden, he looked at certain plants and said, no, this comes from my country, right? And of course, it was in Uppsala, right? And as part of the conversations that we're having, they're quite interesting stories, but I won't bore you with those stories because some of them are quite political, right? And one of the requirements, for instance, for the CBD is that member countries would actually establish, you know, appropriate legislative, administrative, and policy measures to give effect, you know, to the provisions of the Convention on Biological Diversity. And in our case, for instance, I'll just give an example that you've got National Environmental Management and Biodiversity Act, NEMBA. You also have the BAPS regulations, bioprospecting, access and benefit sharing, you know, regulations. You also have, in terms of the patent application, what is called disclosure of origin. And you also, what we also did was to amend the IP laws, particularly to effect, you know, the disclosure of origin. As you're aware that from Nagoya Protocol, there was no consensus, right? You know, around this. But of course, South Africa went ahead and amended, you know, the legislation, you know, to this effect. There's also protection of indigenous knowledge system, IKS. And of course, there was conversations in our space in terms of how do you effect this? Do you just simply amend the IP laws, or you go for a sui generis, approach? And of course, eventually went the sui generis approach. We have you know, specific laws, a law that deals with IKS, and of course, you also have a system to ensure, you know, there's access to genetic material, licensing systems, and mutually material transfer agreement, mutually agreeable terms. I'm sure you're familiar with these terms, right? Show of hands. Thank you so much. So that, well, there's not a lot. Okay, all right, but we'll talk about that. There's a time again to talk about those terms, and of course, benefit sharing. Just to say what was, you know, there's this CBD and all of these things that, you know, the CBD required the member countries, member states to actually, signatories to that CBD to actually, or parties, Congress of, you know, parties to actually be able to, you know, to put in place. And of course, to a certain extent, you know, in the continent, you know, there's, you know, some countries have been able to do this, but others have probably have not been able to do this. But of course, the crucial aspect around this is the enforcement, ability to actually enforce, you know, some of this. And of course, there's always a link. There's a link in our space. South Africa is the third. Get this right. South Africa is the third most biodiverse country in the world. South Africa is the third most biodiverse country in the world. That shows that there's so much stuff that perhaps maybe we have not explored. You know, some cures that are lying somewhere. And of course, it's very important for us to be able to protect and harness that, you know, towards what is always referred to as the bioeconomy, right? And of course, the biotech sector, you know, in terms of commercialization, there are so many opportunities, you know, in there, you know, from in the terms of the pharmaceutical industry, you know, talk about potential lead compounds that are produced by plants that are then used as the foundation, you know, for cures, you know, foundation for medicines, the foundation for, you know, things that actually, you know, solve some of our challenges. And of course, you know, they also on the industrial biotech side could be enzymes for use in textiles, you know, food, feed, detergents, and so forth and so forth. Agriculture, the way, you know, for, you know, some of these tools that are currently in use, you know, insert genetic traits, you know, to improve efficacy and performance. So we're sitting in a chemist, right? Perhaps maybe some of it unexplored. I'll tell you a story. I remember very well in my previous, I always tell this story, right? Because it happened just after the engagement on the IKS. I was in a different institution then. I won't mention it by name. We got approached by a company that was based in Scotland. And that company was actually working on a plant that originates from South Africa. And they're working on a plant, and they then wanted one of the researchers in that institution to actually do some studies on that plant. And of course, we're quite aware. When we then investigated, I discovered that this is a shell company. Right, it's a shell company. It's a front for another company. I wish I wouldn't mention the name. And of course, we had to deal with it, from that perspective. But there are attempts, to actually, bypass, particularly, these CBDs. So what are the issues really when it comes to consideration? To the commercialization of this. I don't know how much time I have, but I can go on. Right. So I thought I'll put, I thought there's so much to talk about, Then let me shorten my presentation rather and talk about things that I thought, were quite important, particularly in the context of, some of the initiatives, that we're working on, as they relate, for instance, to some of the CBD sort of and other. I mean, I'm not going to talk about Nagoyas and all the other things, I think I'll shorten that conversation. Otherwise, I'm afraid that I'll probably be preaching to the choir, right? So one of the important considerations, in my view, is around data ownership and of course, access, to that data. Who owns the data? How is the data, accessed, right? And of course, very interesting article that I came across says, why do people feel comfortable, for instance, depositing their data, deposit their information in data banks, right? Gene banks, right? And of course, there's a certain element of trust. There's a certain element of belief that the data that is put there is safe. But also in our context, I think that maybe there's something else. There's a sort of lack of those kind of facilities, locally that are available. As a result, we probably have no choice, but to use, others elsewhere, right? So, and I think this one is my view, in that if you store data, in a data bank, for instance, or gene bank, it doesn't transfer ownership. It should not transfer ownership. Ownership retained, the person that owns the data, stores the data, should retain, that. And of course, you need to talk about different levels of access. Talk about different levels of access. And the reason why I'm bringing this up is because this is a matter that has also been raised by the CBD in terms of, not explicitly, but it talks about making provisions for instance, for access to data for research purposes. So this is what I refer to when I talk about non-commercial uses, that you actually are able to follow through and see who has access what data, who has access what samples, for whatever purpose that they want to do. And of course, if there's any benefit, any benefit that accrues from that, that is derived from that, it then, accrues, to the people that actually own the data. And of course, and this is very important because in of that link between the indigenous genetic resources and traditional knowledge, right? With regards to perhaps permits and licenses, right? So it's actually very important. It's in line with in our case perhaps, but also very much in line with the CBD, to ensure that when you access resources, you get you have the necessary permits, collection and export permits, particularly from, there's something else as well. I know before I've sort of come back to this one. Very for a fact that most countries, particularly our BRICS partners, India, China, they've got very huge databases for IKS. We also have a database for IKS. It's very important that we place this information in those databases and not necessarily put it in the public domain. Well, eventually it gets to the public domain in a sense, but because we're going to publish the stuff, but make sure that at least there's some form of record of this traditional knowledge and the link between this traditional knowledge and indigenous genetic resource. And of course, in our own space, there's a requirement, when you collection permit or export permit across boundaries, across borders, that you're able to do this. And of course, there's those are the terms again, MTA and MET and benefit sharing. material transfer agreement. When you want to enter into an agreement that you are going to share, materials with another party, you enter into what is called a material transfer agreement. Very clear in terms of what the uses are going to be, and of course, you're not necessarily transferring ownership. And you also indicate if there's anything useful or commercially useful that comes out of that, how is the benefit, going to be shared, by the parties. And of course, the mutually agreeable terms. Usually, when you collect, samples, that are sort of linked to indigenous, traditional knowledge, and of course, these are called mutually agreeable terms, where you're talking about the use, the access, and how the benefit is going to accrue. And of course, very important, we talk about benefit sharing. There's got to be some benefit. There is a plant. I forgot this name. I forgot the name in English, but I know it in Xhosa. It's called umhlonyane, right? Which we use. And there was a company that came, a German company, that decided after some time they made a cough mixture, based on that. And of course, they had filed a patent application. The patent application was actually rejected in Europe, and it was rejected after intervention by an organization called very strange. It's called Paris Convention, not the convention. There's a Paris Convention, but the name of the organization is called the Paris Convention, where they intervened, and of course, to say that this is based on knowledge, on traditional knowledge, that is usually found in the Eastern Cape. And of course, the Eastern government was involved in that, and of course, eventually they reached a deal later on. And also you talk about, some of the requirements in terms of research phase, commercialization phase, and of course, that are sort of in the CBD, and of course, when filing a patent, there's also an expectation of a disclosure of origin. These are some of the consideration, and I think, this is the other aspect that I think is quite important. There's an association of university technology managers, and there's a second one. There's a second point. I forgot responsible licensing. I forgot to include this aspect here, but let's talk about this one now. 9 points to consider when licensing a technology, I'll just go through a few, and I'll skip to the last one, which I think is important, that we consider retaining the right to practice the invention, and also for, other public entities, to have access to that invention. So it's very important. And of course, when you're structuring this license agreement, you want to make sure that the benefit accrues to the greater, accrues widely, to the people that actually require the solution. So you structure them in a manner that promotes, tech transfers. In some instances, companies may want a license only to bury, the technology, right? And of course, it says you prevent reach-through provisions, very important, particularly for material that is linked to traditional knowledge. Basically, you're licensing stuff that doesn't belong to you, that is in the future, and it doesn't belong to you to begin with. That is always, problematic. But I will skip the last two and go to this one. And strange enough, out of the 9 points to consider, is placed last, which I think, in my view, is probably quite important from a commercialization point of view. Include provisions that address unmet need, very relevant in our own space. All right, when we licensing out these technologies, such unmet needs, such as those of neglected patient populations or geographic areas, giving particular attention to improve therapeutics, diagnostics, and agricultural technologies for the developing world. It goes back to the first aspect, why we're talking about tech transfer, particularly within the public, institution space. Why is it so important? It's precisely for those reasons, that the knowledge that we have in our society finds expression in society, and we guard against, as the tech transfer professionals, we make sure that we secure, rights for the communities. Right. I think that is my slide. That is my slide. That is the last one. Is there a question?
Thank you for the interesting talk. I think this is quite important. Maybe next time he deserves 1 hour. I was telling him actually that he's going to get carried away,
I
which
did
he
not.
proved me right because he went beyond the 30 minutes that he was awarded. But I think this was quite interesting to wrap up the day. We still have one more talk. Yes, okay. Closer to wrapping up the day. So I'm gonna open the floor for only 2 questions. I'm trying to be considerate of time, but we can still engage with Mr. Norman. Okay, Olivia, you can go ahead.
Thank you, Ayanda, for the presentation. I'm going to pose this to Erich and Claire, because I want to understand maybe from your perspective or from your institution, how do you deal with the issue of IP, especially now that you having this data in a public access at the end of the day. You have done sequence for more than 200 species. How do you manage that? And how when it comes to access and benefit sharing, and especially because these are the samples sometimes we get in the community. Now you sequence it, and then you want to, at the end, you want to put it out there in the public. How do you engage on that? And I want to bring it to Ayanda, because this is, as the institution also is struggling, we're trying to find a better way on how to do that.
Yeah, I can give you an answer and then hear your thoughts. So yes, we are sequencing lots of genomes from our own lab, from labs that people send samples to us in the United States, as well as from here on this continent in Africa. And at least the IP in the US is that you can't patent natural DNA sequences, regardless where in the world that sequence came from. Whether it's the United States or somewhere else, you can't patent something natural. But you can take the DNA sequence that you sequence, modify it, and then patent the modified version of that sequence for some drug or something else. And that can be done by the person who generated the sequence can do that, or anybody else in the world that has access to that data. The other rule is, typically with NIH funding, you're required to make the sequence public for anybody in the world to use, not just the United States. Where the benefits come to the researchers like myself is the more high quality data we produce, the easier it is to raise money from funding agencies to generate that data and share it. So that's the direct benefit. I don't get wealthy on sequencing DNA, but other companies do. And so we are sequencing some DNA sequences for, like a company, Paratus, for bat genomes, and they're going to take some of those sequences and modify them, but not us. So the benefit sharing is indirect for an academic scientist, unless I do some more patenting. Okay.
So I'll share a little bit of a different perspective from the agricultural side. And our experiences have varied widely with how this has been done, and it's depended on who has funded the research. So the International Bovine HapMap project was actually funded by the producers themselves. Each producer contributed $1,000, and that was how we got the project sequencing done, and also the development of the first SNP arrays. What that meant was we had strong material transfer agreements in place where we had to anonymize the samples. They didn't want anybody to be able to go back to their elite animals and use that information. And so we've come, I think, further forwards in our relationship with producers, and recognizing that there is mutual benefit now of phenotype as well, sometimes being in the public domain, but that's the biggest sticking point for us on the agricultural side. They don't mind us working with the DNA, but they don't want us to release the actual identity of the animals because we are threatening their own development.
Can I add to this? Yeah, so to add to that, there are some companies or some initiatives, they tend to be more private, but with human genomes, they can be with public money as well, where the DNA is sequenced, let's say, of a number of species in Africa and kept behind a firewall. And then they make people pay to access those DNA sequences. And the usual people that will pay to access those DNA sequences is commercial, not academics. Academics aren't used to paying for, such sequences. I mean, maybe USDA is a hybrid there, but and in that regard, if somebody else can come along and sequence those same species and make it public, and then sort of go around the access. But if they don't have the money, then they won't be able to. So.
Can I just follow up? Sorry, Jesse. I just want to. So, when do you release it to the public? Like now, you're sequencing it, né? Then you busy assembling it. When do you release it? After you submitted it to NCBI, or you do that before?
Who are you asking, Eric?
Eric.
Oh, I was asking.
I thought you asked. Okay. Yeah, for these large scale projects like the Vertebrate Genomes Project, we make the sequences, let's say, publicly visible online right away in GenomeArc, as whenever they pass good quality control checks. And then we put it in NCBI, but we have an embargo for use of that data for publication, but still not commercial use. Anybody could still use it commercially as they wish, as soon as it's in NCBI. When it's in GenomeArc in our own SW3 bucket for Amazon, our general principle is people can't use that for commercial use, not until it's in NCBI.
Oh, okay.
Yep.
Okay.
Yeah, so I just want to comment on this aspect. Maybe two comments. Maybe that may explain some of the differences, some of the discomfort. One, for instance, the US is not a member of the CBD. Not a member of the CBD. It's not a member of the CBD. Yeah, no, it's not a member of the CBD, but they get invited. They always address the Congress of the parties, but they're not a member of, they're not bound by that. So in essence, the example that I was giving, the company was a US company, but coming via Scotland, but it's fine, right? So, but also in our context, there's always this link between genetic resources and traditional knowledge. That further complicates issues, and perhaps maybe it may require not a straightforward approach like you, where you just place things in the public domain, but it may require a rethink, around how you then do that, because at the end, you need to give recognition to the holders of the traditional knowledge, and there's another slew of legislation that deals with traditional knowledge. I just want to make a comment around that aspect.
Okay, thank you.
Sorry, I know the US at some point in time used to patent genes in the era of Fenter, but the USPTO then reined that in after a while. But yeah.
Okay, thank you for that interesting conversation. We have another hand at the back.
This is not a question, but I'm not sure if everyone is aware of the Nagoya Protocol. The Nagoya Protocol is meant.
Yeah. Yes.
Sorry?
Yeah.
The Nagoya Protocol is actually meant to address all these issues that you're actually talking about, as you say, except that the US is not signatories to it. So they can attend the meetings, but they're not signatories, so they do not have to comply. But the next COP28, I think, there will be actual discussions on how to implement the Nagoya Protocol and what is meant by access and benefit sharing for the Nagoya Protocol. It may be that there's going to be an international fund that will be available, that everyone's required to put benefits into an international fund that is then shared with the country of origin. That's what the proposal is for the Nagoya Protocol, in which case it makes it easier for you to actually share the information, provided if there's commercial value, that's put into an international fund that is then shared with the origins of the genetic resource.
Yeah, no, let me just comment. No, I'm familiar, with the. I didn't want to discuss that, but I'm also aware of the advanced text, but it's just an advanced text at this point. It's just a proposal, so I didn't even want to raise the issue here. Thanks.
Yeah, just saying something. My own personal opinion about the Nagoya Protocol in its current version is that it's not written in a way, or the policies are not good enough in a way to protect the people they're trying to protect. It's actually harmful, in my opinion, because the protections are so high that people could not do much to actually make benefits of the genome sequences generated. So I think it needs to change in order for the people to benefit from it. And a monetary fund, I think, is one good step in that direction.
Yeah, maybe one last hand from that side, then we'll go to the last speaker.
More a comment than a question as a commercial representative in the room. Intellectual property can be a tightrope that one has to walk, because it can hinder innovation as much as drive that innovation. And I would encourage everyone in the room to basically treat the discussion of IP protection as much as you might consider discussions around publication. A simple metric to justify that statement: if I look to UNISA's records, I think UNISA has about 70, 80 patents. My alumni of the same age as UNISA has in excess of 1,500. My company that I represent has close to 10,000. And that gives you both a measure of the responsibilities involved, but also the opportunity and the value that can be derived for both research, commercial, and government entities.
I would comment on those numbers, but I would rather not, because those numbers, for me, number of patents, I consider to be, I'm not commenting.
Olivia, we said it was the last question.
Yeah, can I just comment one bit? So, no, I just want to bring it. The reason why I'm bringing this question is because we have just started with this sequencing part. And most of the species that we are sequencing are from the community that normally they want to keep their information there. But then my question is, I'm asking because we are now academics, we want to do research on these genomes, isn't it? So I'm coming back to you, Prof, to say, maybe if we look at the Nagoya protocols, and as how Eric said, or on the other hand, it harms us a bit because it actually closed up. Né? But how do we then deal with our issue here in Africa, because we are part of Nagoya as members? So how do we deal with that now when we say we sequence, we release it to the public? What are the challenges there in terms of that protocol?
May I suggest that we take this outside this meeting, because we're not really doing good in terms of time. I know that this is an interesting, talk that it's necessary for most researchers, upcoming researchers, in the different spaces that we're working in. But unfortunately, because of time constraints, we have to move to the next speaker. Okay, is it online, Bongo? Okay. Are they gonna set it up? Okay, thank you. So the next presentation will be from our young scientist, M. Bekai. I had to first check what is this acronym PGPR, because we have different acronyms in our different spaces. So it's harnessing plant growth promoting rhizobacteria for agricultural growth.
Sorry, Mr. Bekai, there's no audio coming from your side. Could you please just check your inputs, please? If you could maybe just check your mic volume, because your audio is unmuted, but there's no sound coming through. So I don't know if you are succeeding, but could you please just check your system sound, your system's mic?
And now?
Yes, we can hear you now. Could you just please share your slides again?
You can see the slide?
Yes, thank you.
Thank you. Okay. Good afternoon, everyone, and thank you for this opportunity. My name is Maseki Bekay. I am biotechnology PhD student at Hawari Boumedi University of Science and Technology. My presentation is focused on harnessing PGPR agricultural growth. We will explore the effect of bacterial strain on plant under salt stress condition. Before diving in, I'd like to give you a quick overview of what we will cover today. Here is a look at the agenda. We will begin with introduction, effect of salt stress on plant, PGPR, material and method, result and discussion, and finally a conclusion. Plant growth is strongly influenced by various abiotic and biotic factors. Soil salinity and the scarcity of water resource are the major factor limiting plant productivity, especially in southern region, not only in Algeria, but in Africa and in all the world. To manage salt stress and its effect, agriculture often rely on chemical products and fertilizers, which over time can negatively impact the soil and its component. Organic agriculture, now incorporating biotechnology practices, has become a key approach. It employs beneficial microorganisms like PGPR, which boosts plant growth through multiple mechanisms, including biofertilization, biocontrol, bioremediation, and biostimulation. Effect of salt stress on plant growth or parameters. Salt stress affects plant growth by impacting multiple parameters processed at various levels. Biochemical changes trigger oxidative stress, altered metabolism, high potassium transport to shoot, lower nutrient uptake, accumulation of abscisic acid and proline, increased antioxidative enzyme, sod catalase and peroxidase. Morphological changes, pool root, leaf rooting and during chlorosis, stunted plant growth and allow high reduction of plant biomass and seedling numbers. Physiological changes, decline or inhibition of photosynthesis, stomatal closure and decline in transpiration, decreased water content, high osmotic concentration, low osmotic potential, and loss of turgor, restricted leaf area and stem length, restricted cell division and elongation. Finally, biomass, grain and yield, sterility, less grain weight and yield, low harvest index. In addition to this morphological, physiological, and biochemical changes, genetic response are manifested notably by the increases in the expression of stress-sensitive gene. PGPR. Plant growth hormone T-chrysobacteria are a derived group of beneficial bacteria that colonize the rhizosphere and enhance plant growth and health through various mechanisms, such as enhancing nutrient availability, protecting plant from pathogens, and stimulating plant growth directly. There is several types of PGPR depending on association mode. Free-living PGPR, symbiotic PGPR, and endophytic PGPR. Rules and effect of PGPR on plant. PGPR exert their beneficial effect through two main mechanisms, direct and indirect mechanism. For the direct mechanism, we have a nitrogen or by nitrogen fixation, phytohormone production, and nutrient mobilization and solubilization, which facilitate and improve nutrition that increase and ameliorate plant growth. For indirect mechanism, by antibiosis, associated desaminase production, lytic enzyme production, and use systematic resistance and siderophore production, which increase abiotic and abiotic stress resistance and protection against phytopathogen. All this enhanced yield. The first part is concerned to bacteria strain tested. The bacteria strain was isolated from rhizospheric soil of Halocnemon, a halophilic plant native to highly saline environment of southern of Algeria. The strain was tested for their PGPR3, with salt tolerance in several concentration of NaCl, enzyme activity, protease, cellulase, amylase, hormone production, especially EII, phosphate solubilization, production of siderophore ammonium and biofilm, associated desaminase, and nitrogen fixation. Among a large selection of isolated, characterized, and identified bacteria, two strains were chosen, BSSM 328 and the BSSM 27. For a long time, these two strains were considered to be a part of the same genus. However, according to Sanjaz Puro et al., they were reclassified into two different genera, Halomonas and Kochneria. Therefore, we selected these two strains to study and compare their effect on plant under salt condition. As we can see on the table, both bacteria shows a positive result for all the tests. The pictures shows some of this result. First one, enzyme activity, protease, amylase, and cellulase. The fourth one is solubilization. Next, siderophore, ammonium production, hormone production, and biofilm. Discussion. According to Itisami and Glick and several other authors, endophytic bacteria and rhizospheric isolated from plant can tolerate a wide range of salt concentration, making them a valuable alias in saline environment. Molecular synthesis for survival. These bacteria synthesize protective molecules such as exopolysaccharide and biofilm, which help them survive under extreme conditions. This substance also provide a protect effective barrier for plant mitigating the harmful effect of salt stress. Nutrient solubilization. These bacteria enhance nutrient availability for plant by solubilizing essential nutrients like phosphate and nitrogen. They also produce siderophore, which bind to iron and make it more accessible to plant. Phytohormone production. Endophytic and rhizosphere bacteria produce growth promoting hormone like indole acetate acid, EII, which stimulate root development and improve plant growth. Finally, enzymatic activity. These bacteria secrete hydraulic enzymes such as protease, cellulase, and amylase, which degrade organic matter and enhance nutrient uptake by plants. The second part is assessment of plant growth promoting ability. After surface sterilized of seed and preparing the bacterial culture, sterilized seed were coated with the bacterial mostly production by the two strain. The experimental design, as we can see, there is a 3 block, a control block and 2 block for the 2 strain. The first line was watered with the distilled water. The second line was watered with an NaCl solution, 100 millimole of concentration. The last line was watered with an NaCl solution, 2000 millimole of concentration. Plant growth was assisted by measuring the following parameter for each plant: shoot root length, shoot and root length, and white, fresh white, and dry white, and dry wheat. And tolerance to salt stress is estimated by calculating the salt tolerance index, STE. For the physiological and biochemical parameters, relative water content was measured, membrane integrity, proline content, total antioxidant capacity, pigment analysis, total chlorophyll and carotenoid, phytohormone analysis, abscisic acid, jasmonic acid, and salicylic acid. The result, the plant growth, the result indicate that salt stress significantly impair various growth parameters. On the other hand, inoculation with both isolate, enhance, and promote growth parameter, both in absence and in presence of salt stress. For membrane integrity, represented by electrolytic leakage and malondialdehyde content, MDA, as we can observe that salt stress, increased electrolytic leakage, and MDA content lead to decrease of membrane stability index. Contrary ways, the inoculation decreased electrolytic leakage, which electrolytic leakage and MDA content, which lead to an increase of membrane stability index. For proline and total antioxidant capacity, the same observation. The salt stress increase these content, and inoculation with the both strain decrease them. The result of a photosynthetic pigment shows that salt stress affects negatively total chlorophyll by decreased their content, and accrete the carotenoid content, which are considered as an antioxidant. But inoculation reverse the effect of the salt stress by increased and ameliorate total chlorophyll and decreased carotenoid content. Finally, the result of analysis of phytohormone content, as we can see and as we can know, abscisic acid play a primary role in acclimatization to osmotic stress through regulation of stomatal movement and limitless transpiration and water loss. That's why we can observe in presence of salt stress, abscisic acid increase significatively in plant with jasmonic acid, and inoculation with the two strain decreased the content of the two hormones. For salicylic acid, who have the inverse effect of abscisic acid and jasmonic acid, have a several content according to concentration of NaCl and type of inoculation. Inoculation with the two bacterial strain retained in this work improved the growth of wheat in the absence of stress, but also mitigate the depressive effect of salt stress. These results are in perfect agreement with finding of El-Bedawi et al., who reported similar results after inoculation of wheat with strain related to Halomonas. The mechanism by which the PGPR improved plant growth are multiple. It should be noted that the two strains used are capable to solubilize inorganic phosphate producing exopolysaccharide and crucial phytohormone, which is EIA. Several authors have shown that the genera Halomonas and Kochneria are PGPR characterized by EII and EPS, exopolysaccharide production, acetyldesaminase presence, siderophore, and phosphate solubilization. Conclusion and perspective: the bacteria treated in this study not only enable them to survive in harsh saline conditions, but also improve plant growth and stress tolerance by enhancing nutrient availability and production growth promoting substance. This study demonstrates that inoculating durum wheat seedling with significantly elevated salt stress, this bacteria enhanced root growth, protected photosynthesis system, improved water status, and stabilized cell membrane. Kochneria showed the most effect in this experimental. More detailed molecular studies are needed to uncover the mechanism that govern plant bacteria interaction and to confirm how this interaction influence and modulate gene expression in both organisms. Understanding the molecular pathway will provide deeper insight into the mutual benefit and adaptive response involved. This is our team in laboratory of soil biology. Thank you for your time and your attention.
Thank you, Ms. Bekai, for an interesting presentation. I think your study just confirmed the importance of looking into the different associations of plants and microbial endophytes and rhizobium bacteria, which are in soil, and the need to further investigate such. I'm going to open the floor for one burning question. Otherwise, I believe everyone is linked to the chat, and then they can post their questions there. One burning question. If we don't have that question, we won't sleep properly. Let's hear that question. Okay, it doesn't seem like we have a question. Based on that, I'm going to close this session, and thank you for everyone who managed to stay until this time. I think it was an interesting first day of our workshop, although we started with a bit of challenges, but I'm glad to see that there's still a lot of people in this room, which is quite appreciative. Without wasting time, let me wish you a lovely evening. And I'm not sure if there are any announcements. Oh, I think they have some lovely presents for our visitors. You're gonna hand them, right? Yes, go ahead. Okay, so these are for the two invited speakers. If you can come, please, we'll really appreciate. Yes, they've already gave a talk today. So I think this is an excellent way to close off the day. Round of applause. Thank you. I think this brings us to the end of today's session. Have a lovely evening, everyone.
Hey there, you're such a tiny thing, but you brought great to the world. Your story became much bigger than we ever dreamed. People lovingly call you Epi, and you changed how we work in the lab every day, more than once. With every little drop that you've held, great inventions could be made. You like it cold. You like it hot. You like it shaky. You're colorful and convertible, and your family grew bigger. There's nothing that can harm you. And that's good, because you preserve for us what we adore most, what's everything to us, the sample.